bensound.com.
Oh, hello everyone. I really thank you for the first day of the event, and we are going to start the second day, and we are going to start with Professor Claudia Vargas. She has a PhD in biological sciences from the Federal University of Rio de Janeiro. Her postdoctorate was held at the Instituto de Sciences Cognitivas in Lyon. She is a full professor at the Federal University of Rio de Janeiro and has experience in neurophysiology and plasticity of the human motor system. The main research focusing on motor cognition in interactions between emotion and action and plasticity in the motor system after central injury. Currently, she coordinates the nucleus of research in neurosciences and motor rehabilitation, headquartered at the Institute of Neurology, Deolindo Couto at Federal University of Rio de Janeiro. Today, her talk has the title, Electrophysiological Fingerprints of Plastic Change in the Sensory Motor Cortex After Peripheral Injury. So, thank you, Professor Claudia, and you can start your presentation.
Thank you very much. Can you hear me? Is it okay? Thank you, Vito, for a nice introduction. I'd like to thank the kind invitation of Professor Baffa and the organizing committee for having us, my team and myself here today. And I would like to say that it's for me a pleasure and an honor to be part of the NeuroMat team, which was first led by Antonio Galvez and now is under the coordination of Osvaldo Baffa. So let us start with work that many of you already know. So since the late 19th century, we know that there are maps of the body in the brain, right? So this, the first evidence, systematic evidence in humans was brought by Penfield and his collaborators, Rasmus and Baldry, and they all have worked with patients who had intractable epilepsy. And what they did was in the surgical theater to stimulate the region around the central sulcus to detect regions that would evoke responses, either sensory responses in the post-central gyrus or motor responses in the precentral gyrus. And this led to the concept of homunculus that I'm sure that you are acquainted with. So until the late 80s, it was there was this central tenet in neuroscience which stated that these representations were fixed in the adult brain. But a series of experiments done in primates and also afterwards in humans challenged this view of fixed representation in the brain. And one of these works was done by Ramachandran. Ramachandran worked with patients who had upper limb amputations. And what he did was to stimulate the face and the stump of these amputated patients and show that these stimulations evoked phantom sensations. So these patients reported the feeling of touching the missing hand, right? For the first time, Ramachandran brought a model where so this is a section of a human brain, depicting a human brain, and the proposal was that the region that was once occupied by the hand was now colonized by upper limb and face representations, right? So this was very exciting at that point. And at the same time, Leonardo Cohen at the NIH has done a TMS experiment in upper limb amputees. What he did was to record on the deltoid muscle bilaterally, and here is the cartoon of his this paper published in 1991. And what we see here is the MAP response that associates to the missing arm to the right and contralateral to the missing arm. And what we can see here is that there are beautiful MAP responses that span across the primary motor cortex, whereas no response at the same intensity could be evoked in the motor cortex contralateral to the sparred arm. So this was also taken as an evidence of change in the MAP's representation after an upper limb amputation. In parallel, several other experiments have been done in primates. So this is a work by John Cass and collaborators, published in 2000. And here we have another technique, which is the microstimulation of the motor cortical surface. Here in the healthy monkey, so we can see here the region that corresponds to the elbow, wrist, and digits, and more laterally to the face and tongue. And after a traumatic amputation, they recorded the motor cortex of these animals and showed that there was this expansion or invasion of the stump and face representation in the region that corresponded originally to the hand. Interestingly, in humans, if you stimulate the region that corresponds to the stump muscle, so this was done in Lyon several years ago with my colleagues Catherine Mercier and Karen Rayleigh in the lab of Angela Sirigu in Lyon. And what we did here was to map the stump muscle representations in a patient. So this is a representation of the patient here. So you can see in the primary motor cortex the map of these muscles with a color-coded sequence here. So higher responses in red and green and lower response in blue. And the dots in red correspond to the places in the motor cortex where it was possible to evoke a phantom movement, the sensation of a moving hand. And what we did here was interesting. We did a dose response curve, as Bafa was mentioning yesterday. We should start to try these types of protocols in humans. What we did here was to record the MAP responses in the stump muscles, biceps and triceps, and to correlate with a scale of sensation of the movement, of the moving hand. So we got a nice dose response curve, right? So the higher the MAP response in these stump muscles, the higher the sensation of phantom movement for these patients. So up to now, what I brought to you was that cortical reorganization, as seen by electrophysiological methods, correspond to a scheme that where before amputation in a healthy patient, the sensory motor cortex has a topographic organization, which follows the classical model of Penfield, and with some improvements, as Vito said yesterday, but the raw schema would be this one. And after upper limb amputation, the region that originally corresponded to the hand is now colonized by face and arm or stump representations. So we could think that this is a field where everything is well established, but last August, Tamar Makin and her colleagues published a very interesting work in Nature Neuroscience, and where she discusses the idea of reorganization after an amputation. So what she states here is that there is stable cortical maps before and after arm amputation. And what is the difference in terms of protocols? The difference is that, so this is a beautiful work. She has done a longitudinal study where she followed patients who had planned upper limb amputations, and she did several measurements before and after the surgery. This is a cartoon showing the patient reporting, or the reports of the phantom upper limb. And she did a 3 Tesla fMRI study where she asked the patients to move the hand before amputation, and after amputation, the instruction was to move the phantom hand, right? And she also asked the patients to do some lip pursing, because she was interested in looking at the activations in the brain for these two tasks. And what she found and what she argues is that there is absolutely no change before and after amputation, which challenges somehow all the work which has been done in mapping, electrophysiological mapping. So this is interesting, and probably I would like to hear your opinion on that, but my conjecture is that the fact that the patient has to intentionally produce a movement, even if it's a phantom movement, somehow produces an activation in the regions that were originally occupied by the hand. And crucially, she also states that there is no change in the movement of the lips. Yeah, sure, please. Take the other one here, Vito.
Well, I can just talk.
Yes, please.
These are statistical maps.
Yes.
And what it means, even if there's activity somewhere, but if it's not, doesn't happen every time, then the statistics will not show anything. So that is one possibility. I don't doubt the quality of the study. I haven't read it, but fMRI is fundamentally different from TMS. TMS we can activate reliably, and also in these tasks, but these are statistical maps. So even if you have activity, it does not necessarily show.
Yeah, thank you, Risto, for your nice comment. Yes, this is the other option will be to, put together these two techniques and discuss on the possibility and limitations that each of them bring to this field, you see? So this is my intention here.
Yeah, Can you hear me? Yeah, I agree with Risto. I think these are different modalities because in fMRI we look at the correlation. Even if there's activation, that doesn't mean that they are like physically connected. So it's different kinds of information. And I think with TMS we are doing at the causal. It's more like corticospinal excitability, and sometimes we use effective connectivity. So it's different approach. That doesn't mean, yeah. So it's different information. Yeah.
Yes, I totally agree with you. The question is that she is claiming that there is no change in the brain after an amputation, which is, and with these longitudinal studies, and she's publishing in Nature Neuroscience, so in 2025, so we have to consider all this, right? So this, but it's a provocation, in fact. Yes, and also, yes, let's go back to this afterwards. Yes, so this, of course, challenged me a lot because we did a study in the with the team of Angela Sirigu in 2009, where we did the mapping of the upper limb and intrinsic hand muscles representation, and also the face longitudinally just after a hand transplant, right? And what we had, what impressed us was first the fact that the hand, intrinsic hand muscle representation could be retrieved in the primary motor cortex from 10 months on in these patients, but also we were very struck by the fact that there was a shrinking of the representation of the biceps muscle and also of the zygomatics. So there is something in TMS that can be revealed, but that is masked or absent in fMRI, as we were discussing here, right? So this is a video just to show the performance of this. I apologize for those that have already seen this video many times, but it's a beautiful video to be seen, and I think, tá aparecendo, não?
Não.
Eu acho. Tudo bem. So this is a video taken 4 years after hand transplantation. Não está funcionando. Não, tem que sair da apresentação. Sorry for this. So you can see here the transition between the transplant here, right? So these patients, in fact, had bilateral hand transplants, and this is 4 years after the transplantation. So you can see how marvelously, of course, with a lot of physical therapy and rehabilitation, these patients could be able to perform hand movements with better than me, in fact, for this kind of task.
Claudio, was that one who became left-handed or not?
Yes, he changed a little bit of his hand preference because for several reasons we don't know well if it was due to surgery or to the fact that the reorganization in the brain showed better results also, right? He performed better with the left hand, being right-handed before the surgery, yes. But is it a question of how left and right hemisphere reorganizes after the transplantation? This is a very nice question to be explored with many more patients.
Because I remember that you mentioned about the preference, lateral preference.
Yes, it can be. We can see in the video that he changes He chooses the left hand
Like
to
an
do the...
ambidextrous, like an
Yes,
ambidextrous.
yes.
Very interesting. Thank you.
Thank you. So for this first set of results, as I said, with electrophysiological methods, you have this change in the representation of the primary motor cortex after the amputation, which is reversed after the hand transplantation. Okay, so this is a question maybe for later, for us in the afternoon, the discrepancies between the fMRI and TMS mapping results. So one possibility could be related to the fact that the participants in the fMRI design are doing phantom movement, so it's an active, intentional activation that is retrieved in the fMRI, whereas in the TMS studies, we are always doing passive mapping, so this could be something to be considered, but also as Risto and Julio pointed out, the techniques are so different, and maybe we can discuss more on that in terms of their performance in revealing the reorganization in the brain after an amputation. So a question for our roundtable in the afternoon, maybe? Okay, so back in Brazil, we had the opportunity to work with another set of patients which was the brachial plexus injured patients, which are becoming very, very frequent in Brazil. We have a huge amount of people using motorcycle. These are young people, and they suffer a motorcycle accident, and then you have this injury in this region, which is the brachial plexus. And depending on the level and the gravity of the lesion, so this set of nerves carry sensory information from the upper limb, from vertical vertebra C4 to C8 and C1, and also carries the motor information to control the upper limb. So there are different types of lesions depending on the gravity of the accident. And our question was, are there any changes in the sensory motor maps after brachial plexus injury? So here again, this picture depicts the basic idea that the reorganization in the sensory motor map, as evidenced by electrophysiological methods, would correspond to these dynamics in this change in the representations in the sensory motor cortex. So we had the opportunity to work with a set of data, fMRI data, which was kindly provided by Martin Malesi from Leiden University. We had the funding for this study from Capes no FIC. And what we did here, this study was led by Daniel Freiman from Buenos Aires. And what Daniel proposed was to analyze the local correlation between voxels in different regions of the primary motor cortex. So here again, the cartoon, the Penfield's cartoon, where what we did was to separate the region of the primary motor cortex in sub-areas, colored here, in, for instance, the trunk in green, upper limb and hand in blue, and the face in light blue. And these regions were then analyzed with a method where we can correlate the voxels among each other, and as you can expect, there is a decay in correlation as a function of distance. Here in dark in healthy controls, and in pink in brachial plexus lesion patients, right? So as a function of distance, there is a decay in correlation. This was resting state data. So it was interesting to see that for the brachial plexus patients, there was a reduced correlation in all the regions that correspond to the trunk and upper limb regions, and this effect was absent for face, for instance. So these are the statistical maps for these effects here. So this was the hemisphere contralateral to the affected limb. So the patients had done a brachial plexus avulsion, which is the more severe case, lesion case. But an effect was also found, as Risto beautifully showed yesterday. We have this corpus callosum connection between the two hemispheres, and it was very evident that for the hemisphere contralateral to the unlesioned limb, we also found decay in correlation as a function of distance for the hands and trunk regions. So this is a first wrap-up of this new set of results. So the unilateral brachial plexus injury leads to reduced functional connectivity at the primary motor cortex in both hemispheres, with no gray matter reduction in M1. I didn't show these results, but this is what we brought in the paper. And we took these changes in the motor cortex functional correlation as an evidence of cortical circuits disorganization. And the next question was, well, is it related to the inhibitory circuits? So we came back to TMS protocol, employing this technique called afferent inhibition. As some of you are certainly very familiar with. So the idea here is to, when you apply a pulse, you have a MAP response, as you know already very well, right? But then if you stimulate the periphery before, some milliseconds before, you can induce a reduction in the MAP response as a consequence of a sensory motor integration in the cortex, which leads to a reduced peak-to-peak amplitude in the MAP response. So this is the basis of the afferent inhibition. It's considered as a marker of sensory motor integration. This is from a review of Turco and collaborators. There are two very well-established types of afferent inhibition techniques. In the short afferent inhibition, the electrical stimulus, which can, by the way, be applied in different parts of the periphery, precedes the TMS response from 20 to 50 or 60 milliseconds, and then you have this effect of inhibition in the MAP as compared to a control condition. And for the long afferent inhibition, the peripheral stimulation is applied from 100 or 200 to 1,000 milliseconds before the TMS pulse, and we also evoking an inhibition in the MAP response. So we started by sending our very bright students to spend some time in Karen Haley's lab in Lyon, and she did a beautiful work there under supervision of the colleagues in Lyon, trying to answer the question of this face-hand sensory motor interactions as revealed by afferent inhibition. So as I showed you, the classical fMRI methods are not very suited to show these effects, face-hand effects. If you look at the homunculus, in fact, there is a clear separation, or seems to be a clear separation between these regions, And the question was how this separation is maintained. And one possibility is that the inhibitory circuits are regulating these borders, right? And so what the colleagues did in Lyon was to stimulate the face, upper limb, or cheek, and evoke a map response in the FDI, right? And what is represented here is, in fact, this is done for several intervals, right? So they chose these intervals for the, as I said, short afferent inhibition from 15 to 85, and they found a clear inhibition effect for this times interval from 45 to 65 milliseconds. So as the standard in this domain is to consider the inhibitory effect as a function of the map response evoked without the peripheral stimulation, right? So the graph represents the inhibition as a function of the pure map response evoked by TMS stimulation. So upper limb and cheek, a clear inhibition effect. So we took this paradigm to investigate the effects of the brachial plexus injury in this very border, the border between face and hand, right? So this work was the PhD thesis of Fernanda Torres. This was our setup at that time. And in the lab, now we have a robotic arm in the lab, so things are improving. But we are already using the Invesalius co-registration system, kindly provided by the Laboratory of Biomagnetism here in Ribeirão Preto. And we have to say special thanks to Renan Matsuda, who helped us uncountable times to solve the questions related to the co-registration. So here what we did was to do the electrical stimulation in the hand. So we had a block of testing where we electrically stimulated the finger and recorded in the FDI. And in another set of blocks, we stimulated the face and recorded in the FDI. So this is the type of results I'm going to present to you now. First, for short afferent inhibition, hand short afferent inhibition, and face hand short afferent inhibition. For control, participants in blue and TBI brachial plexus injured patients in gray. So we were able to produce afferent inhibition in the control group in a very classical way. And for the patients, we did have also the afferent inhibition for the hand, but with a little delay. You see that there is a huge variability across the patients, which is always something to consider, as the colleagues discussed yesterday. And for the face hand, what we found was a difference between the control group and the TBPI group, as if there was disinhibition in the TBPI group as compared to the control group for face hand short afferent inhibition. So we took this evidence as related to this disinhibition in the borders of these two representations. For long afferent inhibition, again, an effect in the control group at 100 milliseconds, which was expected, as I said, and a type of facilitation in the brachial plexus injured group for the longer intervals, mostly for the long intervals, for hand-hand, but also for face-hand afferent inhibition. We find a difference between the controls and the BPI, where sort of the inhibitory modulation is reduced, right? So wrap up here, and the unilateral brachial plexus avulsion leads to reduced inhibition in hand-hand and face-hand sensory motor interactions. So these effects are even more evident for long latency afferent inhibition, indicating a disorder inhibitory regulation at a mesoscopic level. But of course, it would be wonderful to do another experiment with TMS EEG study with afferent inhibition to map, as Risto showed yesterday, to try to map these effects, really map at the mesoscopic level, employing the EEG associated with the TMS, right? So we have some work in process to investigate the effects of the temporal sequence on these afferent inhibition effects. Raquel Carvalho is going to tell us a little bit more tomorrow about these results. And how am I with my time? Sorry.
If you go 1 more minute, we have 15 minutes for questions.
1 more minute.
Maybe we can take a few minutes.
No, I'm doing some propaganda, right? Some publicity. So, well, these patients are often submitted to very radical, I should say, and challenging surgeries when the brachial plexus injury is very severe. One of the techniques that are employed by the surgeons, this is our colleague in Rio that have participated in many of our studies here and is also a co-author of this paper, where you take the branch of the innervation that goes to the intercostal muscles and you bring it to the bicep muscle so that you can give back some control of the flexors of the upper limb, right? So there is some study done with these patients and discussion on the fact that there must be plasticity in the brain that associates with this huge change in the periphery. And this is the topic of Anna Carolina Schmeidecki, who is presenting a poster on Wednesday. So she's going to tell you about this very beautiful model of plasticity where you have referred sensation that is induced by this surgery. So she has done some very detailed mapping of these effects where you stimulate the patient's arm and you evoke sensory response in the chest. And she has devised a very nice protocol to afferent inhibition protocol to investigate these effects on the bicep muscle. So she's going to tell you more on Wednesday. We also have a homepage for scientific dissemination on brachial plexus injury, who is supported by NeuroMat from the very beginning, very, very important for us. And this is my team. It's a multidisciplinary team, and I would like to thank your attention. Thank you very much.
Thank you, Professor Claudia. We are going to start the questions.
Thank you very much for the nice lecture. Maybe it's a naive question, but my question is regarding that Nature paper you showed about the neuronal activity after the amputation. I was just wondering that I have been reading some papers that talk about how neuroplasticity changes when people age. And I was just wondering if that was just an effect of low plasticity in the brain, and this could be like an age-dependent study. Different ages with different types of amputation could present or not like changes in the brain functions, because I have seen some lectures that people talk about when we age, our brains like use with that particular connectivities, and even that when you have like amputation, that connectivities could still be present. So I would like to hear comments about that, if this can be like an age-dependent effect or not.
Well, although the paper that Makin has published has appeared as a short communication, in fact, she has a lot of data on employing the same protocol on 26 amputees, and it's a very robust result. Of course, in a longitudinal study, she had only 3 patients. It's very difficult to make a good longitudinal study, and these kind of questions, for instance, aging, are very difficult to control for, you see? You have to have a huge cohort to state or to really be sure that this is an aging effect. So.
Hi, nice talk. Thank you for sharing with us. 2 main questions. The first one, you think that the difference between the etiology of amputation, like no traumatic or traumatic amputation, has some difference in neuroplasticity, and the peripheral traumatic or not traumatic injury is different in this neuroplasticity and the effects? Thank you.
It's an excellent question. Because, of course, we are not taking into consideration the emotional aspects of the accident, but we know from other evidences that this plays an important role in how the plasticity develops, but also the fact that you can have pain associated to the amputation, and this is a major, say, confounding effect, but which is present in most patients, right? And even having or not a phantom limb or a phantom pain is very variable across subjects. So it's a very interesting topic, I should say.
Nice talk, Claudia.
Thank you.
I'm not in the field, but I remember to see a lecture from Raman Chandran regarding this type of amputation. And at that time, I don't know if the hypothesis changed with time, but he mentioned that when you have not necrotic, but pain and body destruction, and I think that pain was a very important effect, that would be contribute to more expression of phantom limb, and even with this mirror things that he would kind of re-educate the organization of the cortex with visual and symmetric. So like the visual input would correct this. Is it still a relevant hypothesis, or it's changed?
Absolutely, yes. I think, I mean, everything for me and for the evidence, people that works with plasticity induced by, for instance, watching the movement of the sparred hand projected towards the amputated limb is a form of therapy which is being used not only for peripheral lesions but also for central lesions, for instance. So it's difficult to reconcile with this proposal of Tamar Makin by saying that everything is stable. I think maybe we are looking at the problem from different perspectives. Of course, there is a residual information there. Of course, our brain is powerful enough retrieve this memory information, which is important, which relates to the fact of actively trying to move the phantom limb, you see, I think and also as was discussed before, the fact that the methodologies are quite different and things that we should think about. No? Thank you.
Okay, thank you for the questions. Do we have more questions?
I know I could ask this question outside, but let me take this opportunity. It's about the thing that Marcus commented. I know that the patient that you showed changed the hand that he used it more after the transplantation, but the pace with which each hand recovered was different also,
Yes.
isn't it? And it was faster in which hand?
In the left hand, non-dominant hand, yes, for both patients.
Yes.
Which is so interesting because it's possible that there are some central constraints that associate with the dominant hand, which precludes a faster recovery for the dominant hand. Yes. Antonio Galvez liked a lot this difference and the idea that there are some constraints in the way the fact that the dominance imposes to the brain, which could be modeled mathematically. This is why he was so excited about this. And then how the fact that the structure is so more hardwired that it would be less plastic. This would be interesting to explore, but you know, we have to have many more than transplanted patients, which are still rare in the, yeah. Thank you.
Okay, more questions? Okay, so I really thank you, Professor Claudia, for the talk. So.
I have a last slide here. Thank you, Vito. Now you have to move. Yes. So now we move to the NeuroMat research snapshots. Will be 20 minutes talk, 25 minutes talks. Paulo, do you want to install? So the first will be held by Paulo Cabral Passos. Paulo has done his PhD at the Institute of Biophysics in Rio de Janeiro and is postdoc at NeuroMat. And he will be telling interesting results on the type of modeling, in fact, that we've been doing in NeuroMat on the last few years with the NeuroMat team. And then Bia Ramalho will present some preliminary results on employing this type of modeling, right? Bia Ramalho is also formed in the Institute of Biophysics, Carlos Chagas Filho, in the UFRJ, is now a postdoc at FAPERJ in my lab. Você vai usar o...
Deixa eu aqui, sorry, I'm just solving some technical details. You know how to... I'm not familiar with this PDF reader.
Me too.
Control. I would like first to thank Professor Baffa for the invitation to give this presentation. Professor Claudia already introduced me. I will be giving this talk along with Bia, okay? And I would like to emphasize that this work is a joint work with this very special team that I have here listed, okay? So I will be talking a little bit about the context tree framework for those who are not familiar with it, okay? And about some experiments in neuroscience that we've been doing along these years in NeuroMat. So first, the motivation behind the context tree models, okay? The motivation for context tree models come from a principle called the minimum description length that was proposed in the late 80s by Hyssinen, which is one of the greatest foundational figures of modern compression theory, okay? And it says that the shorter description of the data corresponds to its best achievable model. And guided by this principle, he, in 1983, published I think his most important paper, An Universal System for Data Compression, in which he introduced the concept of context trees. So the way that I like to explain context trees is always by an example. I think it's the best way, okay? But first, I would like to emphasize that, as I said, Hyssinen worked with compression theory, okay? Compression theory has a very intuitive idea. Most of the sequences in real world, or at least many of the sequences in real world, they grow in length up to infinity, but the information within that sequence is bounded, okay? So this means that most sequences are redundant, and context tree models tries to capture this redundancy in the sequence. So I present to you a sequence that grows in length here, okay, in a very, very small experiment. So that sequence starts with a 0, then a 2 comes, then a 1, then a 0, then a 1, then a 1, a 0, a 2, a 1, and okay. I hope that you capture the structure of the sequence I just presented to you. So here I place a snippet of the sequence, okay, but I place also 2 black boxes, one on the left and another one on the right, and you can just see this one. And I ask you to predict what is the element that comes next, okay? I will talk here with an imaginative person in the audience, and I say that this person remembers the structure well, okay? So if I ask this person what comes next, maybe he or she will say that, I don't know, I think I saw at least 2 situations in which A1 occur, and without seeing other elements in the past, I think I can't say what is coming next, okay? So I present you another element of the sequence in the past. And that person said, no, now I am 100% sure that A1 comes next, okay? And I say, don't you want to see, for example, another elements in the past, like this, or this? And he says, no. The only fragment of the past that I need to see is this one, okay? So this, in that sequence, is what Hyssinen called a context. Is the minimum sequence of past elements necessary with all information necessary to predict the next element. And along with another piece of information, we have all we need in order to compress the information of the sequence, okay, and represent it. So now I present to you the sequence generating machine. Okay, the name, I would like to say context tree model because it's short, but the name is probabilistic context tree model because we have this context in the sequence, so we can have a very big sequence, but a small set of contexts representing them. Okay, but we also have a probability distributions associated with each context in the sequence. Okay, so in this table that I show you, let me pick up the mouse. We see the set of contexts here, and here on the top you see the elements of the alphabet of the sequence. So it's a sequence of 0, 1, and 2. How we read this table? So here we have a context 0, in which in the cell we see the probability of seeing a 1 after the 0. The probability is 0.3. The probability of seeing a 2 after the 0 is 0.7. And the probability of seeing the 0 after another 0 is inexistent in that sequence. And all the other contexts that we see here are deterministic. Okay? So, since these contexts can be arranged in an upside-down tree, where you see them here, okay, in which we have some structures. Here's the root. Here is the node. We call this a branch. This model have been named a context tree model. Okay? So here in the bottom we have another example of the sequence. And in the 0, in which we have a non-deterministic transition, you can see the transition that end up occurring in the sequence. Okay? So in 2013, the NeuroMat was born, and it was led by Professor Galvez in a very special team of science. We have a lot of them here. And one of the objectives of the project was to tackle some conjectures in neuroscience and to provide evidence, and not only provide evidence for these conjectures, but ground this evidence on rigorous mathematical framework. And one of these conjectures was this one from Helmholtz that says that the brain actively and unconsciously perform an inference on sequence of events. This originated the line of research that we know as the statistical brain, which I am part, okay? And the context tree models played an important role in this line of research. So I think I'm not mistaken to say that this was one of the more important papers in this line of research that was developed within this line of research. And the object in this study here was to try to identify that context tree model in the EEG activity of the participants in an experiment, okay? So here in the bottom, you see an EEG trace, okay? And you also see, I hope you see, some elements on the bottom. It's 2, 1, 0. Here is 2, 1, 1, 2, 1, 0, okay? This represents auditory stimuli that were presented to the participants. The 2 is a strong beat, the 1 is a weak beat, and 0 is a silent unit, okay? So we have these stimuli regularly spaced, and in this paper, it was used 2 context tree models. I'm here showing just one to you for the sake of simplicity. And the objective was to retrieve a context tree model from the EEG. So the idea is, if the participant is stimulated with a sequence, the participant must hold in its EEG a signature of that sequence that is compatible with the context tree, okay? So how we do that? We do that by employing a statistical model selection called the context algorithm. And this context algorithm starts with a very special tree that we call a maximum admissible tree, that is a function of the sequence of stimuli given to the subject, okay? And we shaped that tree until the final model that we say, this is the model for the participant, okay? So here in B1, I'm showing this maximum admissible tree, and you see here a branch that is highlighted. You see a 0, 1, and 2, and a 1, and 1, and 2. Both of these subsequence appears in the sequence of stimulation, okay? So we ask ourselves, is the law of the first subsequence in that branch equal to the law of the second subsequence, you must pay attention that they only differ by one element, and this is the last element in the past, okay? And with this null hypothesis, if we reject it, we end up with this branch in the final context tree, and otherwise we prune this branch from the final tree, and we get a tree that looks like this. For example, in this step, we visit each branch, okay? And in order to test this null hypothesis, we use a projective method. So here we have a set of segments of EG, one associated with this first subsequence, another associated with the second subsequence, and we project them into a Brownian bridge. What we are doing here is a dimensionality reduction. And since we have two subsequences, we end up with two distributions that we can test in order to know if the law of the EEGs are the same or not. Okay, we do this with a Kolmogorov-Smirnov usually, okay? But we, as I said here, we project into a Brownian bridge. This is like a reference. We do this with several Brownian bridges. In fact, we do with 5,000 Brownian bridges for each branch that we visit and each pair that we test, okay? So here is a little bit of the results. This is the other tree of stimulation that was used. And we retrieve a context tree for each electrode. So you see here, there are a lot of trees. These trees here are what we call a mode context tree. There is a function of the set of retrieved trees for all subjects, okay? And we see that we retrieve the exactly same structure of the stimuli for some of the electrodes here. I'm not showing everything because I think you can barely see something with this size, okay? But this scale here that you see, once it says that it is the same tree from the simulation A0, it says that no contexts in the retrieved tree are similar, okay? So, sorry, more technical details. I swear I just pressed the button. Finally. So, this is a paper that comes from my PhD thesis. Here we explore a similar conjecture. Here we're not interested in EEG, but in response times from subjects, okay, throughout the game. So here the participants played a game called the goalkeeper, in which he plays the role of the goalkeeper and has to predict at each step, each trial, what will be the kick of the penalty taker, okay? And this penalty taker, he generates a sequence of penalty kicks that comes from a context tree. So we want to retrieve the sequence of stimulation from the response times. And this is a summary of the results. Here we see the structure of the context tree a little bit different, because here we don't see the context. We only see the number of subjects in which these contexts appear. This is for the first block of the game. This is for the second, and this is for the third. The participant played 1,500 penalty kicks, which is a lot of penalty kicks. And we saw here from these mode context trees that from the first block to the second block, we have an increase in the number of subjects in which the retrieved tree contains the context of the stimulation, okay? But in the third block, we see that there is a reduction here in this middle branch. Our initial conjecture was that the response times only was dependent on the context and not another thing. But with response times, we saw that this is a little bit different. What we saw is that after the participant had made a prediction failure, so he thought that the penalty taker should write, and he chose less, for example. There is a kind of perturbation in the distribution of response times in that context. So this was responsible for this reduction in the number of retrieved contexts in this branch, for example. And as we see, when we are dealing with behavioral things, that is a little bit more difficult. In this first paper that I showed you, the participants were only with eyes closed, just listen to the sequence of auditory stimulation, okay? So here I present the last results. This is a work in preparation. I think we will have a preprint like in a month, I hope so. And here we just are doing the same thing that we did on the last paper, but now we are recording the EEG too, and we have very interesting results. So here we have the scalp, these electrodes that we are recording, and here you can see the EEG and the stimulation, okay? We make the same procedure that I just showed you, but we add another layer to it. We calculate the distance from the context tree of the simulation and then the retrieved tree from the EEG. This is a very special distance developed by one of our collaborators, Ricardo Freiman. Okay, and with this we can make some more quantitative measures than we did with the mode context tree that was our last approach. So what we did here is see various windows across the game, okay, and we capture the distance of the retrieved trees. So we can see here a scalp, here we can see a color bar in which we have a distance, and as the participant plays, we see that the median distance reduced throughout the game. And then we made a linear mixed effects model in order to see if we can capture a relationship between the prediction accuracy of the participants or the success rates and the distance. And we see that we have very interesting results. There is an inverse relationship that is what we are thinking we will find at first place. And these are the electrodes in which we have a significant correlation. And we found that the most significant correlation was found here in the parietal area, which is an association area, and so it makes sense. And then we hope we have a preprint very soon in order to share with you, okay? So here I end my presentation and I pass the word to Bia.
Hello, everyone. Agora eu preciso sair daqui. Paulo, como você fez isso?
So the questions will be addressed at the end of the session, right? Thank you.
Aqui, tá? E daí você vai aqui.
Não, o meu é PPT.
Ah, o teu é PPT.
Hi, everyone. Thank you for the invitation, the organizing committee. It's a pleasure to be here with you. And today I'm going to share with you preliminary results we have applying the goalkeeper game. I will talk a little bit more than Paulo about the goalkeeper game. And this work was part of the PhD thesis of Vitor Hugo Moraes, and this is our friends that is working in this project. We are still collecting data, so we are, if someone is from Rio de Janeiro and want to to be part of this project, we are still collecting data. So the project is the goalkeeper game. We're covering the stochastic structure of the sequence of events from motor potentials evoked by TMS. And why we did decide to use the TMS to tackle this topic? Because predictive mechanisms have been studied through the primary motor cortex and the cortical spinal tract. In fact, the motor cortex connects with several areas of the brain, important areas for decision making, and it's somehow the article suggests that what we see in the motor cortex can be something that is modulated by the prefrontal areas or decision making and etc. And the TMS technique has been used for different purposes, and including the physiological change associated with the motor preparation. That is the phase we will tackle in this study. And also the moment in time that Paulo was analyzing his EEG data. One important study that looked at predictive coding in the primary motor cortex was this one from Bestman and colleagues from 2008. It's very different from the one we will show you later, but it's the one that we think that get closer to us. So we may talk a little bit with this work. So in this work, they present a stimulus that was they need to see something, and they need to press a button with the thumb or the pink finger, the little finger, depending on the cue and etc. So there was a cross fixation, and then a symbol. For the thumb press was a square, a red square, and then a cross fixation, and then the TMS stimulus right before the go signal that was the signal to really press the button. So the first one was like a cue that could be a valid or an invalid cue. So in this case, we have a valid cue, so it's a red square, and then the go signal was the red square, so the subject should press the button with the thumb. And in this other case, there was a triangle, and the triangle was the pink finger, the little finger pressing. So this was the cue, but then the go signal was the thumb. So the person maybe preparing to press with the little finger, and then they should press with the thumb. And what was the manipulation thing in here? So they manipulate the number and the probability of valid and invalid cues. So in this case, the 50 to 45 was the number of the percentage of valid and invalid cues. So in this case, there was no possibility of prediction. It was something really random, and the person couldn't learn that there was something that was something that appears more and then a surprise. No. And in the first case, 85 to 50, the percentage of invalid cues was 15%. So there was something that was a surprise, this 15% of trials. And what he see that it's just a little result on the entire project, but when the cue was the thumb, and here in the bars are when the go signal was thumb and the go signal was little finger. When the cue matched the go signal, so the subjects had a bigger MEPs. And when the cue was the thumb, but then they should press the other thing that they was not preparing to press, then the MEPs were smaller. So in this case, they said that the motor output was suggesting that the motor output was biased according to the context probability of the experiment. And so, taking in consideration everything that I've said about the motor cortex and what some people had found, the idea of this project was to investigate, the aim was to investigate the impact of learning the stochastic sequence, in this case of the goalkeeper game, or that is the impact of the context, on motor evoked potentials recorded from the first dorsal interosseous and the flexor digitorium superficialis muscles. The first dorsal interosseous is for flexion of the index finger, and the flexor digitorium superficialis is more related, in this case, the place where we put the EMG to record the activity was more related to the little finger and the ring finger. So the goalkeeper game is, I'll just show you, tava funcionando? So the goalkeeper game is something like this. You are seeing here, this is what the subjects watch. It's a game, a computer game, that the subject has to sit in front of TV, and they have to predict the person, the subject is the goalkeeper, and they have to predict before the penalty taker make the kick, for which side of the screen the ball would go. That is, when the arrows appear in the TV, the subject this was the goal signal for the subject, and then the subject should press to the right, middle, or left, and then after the selection of the subject, the penalty taker kicks the ball, and then there is a feedback. Then the person will see if the goalkeeper take or didn't take the ball. Okay? And this is the instruction they received while initiating the goalkeeper game. So they are goalkeeper, must decide, and etc. Everything I have already said. So the sequence the alphabet that we select was also the 1, 0, 1, and 2 that Paulo said before. And 0 was the left defense, 1 was the middle one, and 2 was the right one. So the sequence of the penalty taker and the sequence of the goalkeeper, and the selected one by the subject, was translated as 0, 1, and 2. So 0 left. And this was the context treat we used. So it's similar to the previous one that Paulo showed, but the one he showed, there was this branch in the middle, and this one we put the branch in the left side. And this is the probability matrix related to this context tree. That is, we have several deterministic contexts, but after the symbol 1, or the context, the past 1, the sequence could be followed by a 0 or a 2, with a 0.3 probability of 0, that is the low probability 1, and 0.7 of probability to have a 2. Obrigada. So this is the example of our sequence, 1, 2, 0, or 1, 0, 0. But the 1, 2, 0 was the most frequent, and 1, 0, 0 was the less probable, or less frequent. Okay? This is the temporal representation of our trial. Due to the structure of the goalkeeper game, we couldn't really change the time we put the TMS pose. So we decided to use the initial moment of the trial. So 400 milliseconds before the appearance of the arrows or the goal signal, we give the TMS pulse. And then the arrows appear, and then the subject could select for which side they want to make the defense. And this was our response time. And then we have a feedback of 3 seconds. So this is a bit different from the previous study, because the goalkeeper game used by Paulo and our colleagues in the previous experiment has smaller trials, so more shorter trials, so less seconds. But to use the TMS, we need to have at least 3-4 seconds between trials. So we increased, we make a larger feedback to have this inter-trial interval a little bit bigger than to choose the TMS. So I'm sorry, I forgot to translate the image. But the subject was seated. The hand was in a keyboard, and it's a special keyboard, and they should put the index, the middle, and the ring finger in the buttons to select 0, 1, or 2. And we use also the Inversaglios navigator to make the co-registration of the TMS. And we registered the motor evoked potentials from the FDI and the FDS, but we select only the hotspot of the FDI. The experiment, it's really big. We have many trials with the goalkeeper game running. So if we stop to change the hotspot, it would take many more time. So we decided to tackle the FDI hotspot. And by now, we collect data from 16 health right-handed male volunteers around 24 years of age, and with extensive or some experience with electronic games. It was approved by the ethical committee. So this is the study design. So we have a familiarization block in the beginning, and then we have blocks of 200 trials of the goalkeeper game, but we intercalate with and without TMS. It would be amazing if we could do everything with TMS, but due to problems with coil problems, it's hitting, so we couldn't run the entire experiment, so we had to give some pulses and then stop to refresh the coil. I guess it's a good... and then we intercalate because of this. And today I represent basically the data with TMS, preliminary data. And we use a context tree algorithm that Paulo said before to retrieve the context trees from the MEP data, but also from the response time data. So this is our first result. So we first look at the success rate. So first is the success rate, average success rate. That is the number of correct responses divided by the number of trials per block. So the success rate is presented by blocks. And what we see is that the success rate increased. So the subjects learn the task, and it is corroborating previous studies, so they get better across the blocks. And when we start looking at the FDI maps, we decided to first, and this is a preliminary, so I would love to hear from you or other ideas. And we decided to separate the trials by the context. So here you have 0, 0, 1, 1, 0, 2, and 2, 0. And for the FDI, we see that there is a difference between the context. but is something that didn't appear at the postdoc test. But there is a trend that after the context 1, 0, the MEPs are higher, bigger, larger than the other context. And the context 1, 0 is the context that appears less because it's a 1 followed by a 0 that is the low probability moment of the sequence. So it appears that after a low probability context, there is a increase or a modulation of the motor output. And when we look at the FDS, what we saw was that after the context 1, and this one has a significance, a statistical significance, after the context 1, that is the only moment that the subject can press with the ring finger, that is the 2, and that is the finger that is more related to the FDS muscle that is used to, is the FDS muscle that makes the flexion of this finger. And this was the moment that we saw an increase in the motor evoked potential. And what we think is that this is more related to the motor preparation to press that button. So it's the only moment that they press with the ring finger, and was the moment that we saw the increase in the motor evoked potential. And when we retrieved the context tree, and here I'm not showing each participant context tree, I'm showing the mode context tree. And the numbers inside are the number of volunteers from whom the corresponding branch was presented in the obtained trees of each individual. So this is the sequence context tree, and this was the context tree retrieved from the response time. So we repeat the data that Paulo showed before. And when we look at the FTI and MEP's context tree, we also could retrieve the context tree from the MEPs. But when we look at the FTS MEPs, it was impossible to retrieve the original context tree. And what we think is, especially due to the variability of the MEPs, it's not like the response time that we have something very, very strong. Maybe it's due to the variability of the MEPs that we couldn't select, but in general, we think that we could retrieve the FDI MEPs because maybe the FDI it's influenced by the probability of the sequence, and the FDS not, and maybe the FDS is more. It's a more general. The FDI makes more fine movements, and there is maybe a more specific representation, motor representation, and maybe the FTS that is flexor and more general flexor of the 4 fingers of the hand. Didn't capture or isn't influenced by the probability of the sequence. And taking I said before, the results suggest that modulations in corticospinal stability go beyond the motor preparation necessary for movement execution, being influenced by the probability structure of the context tree. So we are still planning what we will do next to analyze this data. We are planning to make apply the distances that Paulo said analyzing the EEG data, but we are also thinking about splitting the subjects in strategies, because the subject can decide to go to the more and select the more frequent answer as the two, or the subject can try to match, or maybe next time will be 0, or next time will be 2. So there is this probability strategy difference that might influence the response. You heard more about context tree and physiological data, electrophysiological data, in the short talks. Vitor Hugo and Thais will talk about another way to look at this, but not using the goalkeeper game. And Priscila Azevedo will present a poster in the poster session, the previous study that Paulo said, that the one with the EEG. So I'd like to thank you for your attention, and we are open to questions.
Thank you, Paulo and Bia. Very nice talks. Now we are open for questions, please.
Thank you for the nice talk, both of you. So, yeah, my question, maybe both of you can reply. So, have you think about to compare your protocol, like of the stimuli protocol, with like random stimuli, or even a fixed, like, instead of using like the context tree, you use just 0, 1, 2, and so on. Do you think the results will be clear different? What is your take on that?
I think we can do that with simple experiments, maybe using the response times, because it's shorter to gather data. But there is always also the problematic of adding another piece of the protocol that can influence the subsequent one, you know. Because it doesn't make sense for us to do this for some set of patients. We need to do the same set of patients. Okay, so the participant will play a random sequence, and then he will play the context tree. Okay, I don't know if using a uniform sequence is the best. Maybe a sequence that they cannot learn and compare across situations. I don't know. But the short answer is no. We are not intending it, because
I don't think it's a lot of time to run this.
It takes a lot of time, and can also induce something on the participant that may be influencing the subsequent part. Okay.
Right. Thank you.
Since we have this part of the sequence that is deterministic, maybe I guess we could say something about this, and we say something about this when we compare the MEPs or the response we find, looking after the deterministic part of the sequence and the stochastic part of the sequence.
Maybe you could compare like the learning curve with like these two conditions, or not conditions, but
No, I know. I don't think we can split this. But the learning curve of the goalkeeper game sequence we are using is for the first to the second block they have already learned. No, so this one is the sequence we have is it's already pretty easy, and in fact they learn the deterministic part of the sequence because it's impossible to learn the stochastic one.
I would like just to come back to that part, but we can compare the part where he is learning to the one that he is not. But in that sequence, as Bia said, it is very easy. We wanted to have a easy, so it's a proof of concept. But I think maybe the best way is to compare when the participant is learning to when he already learned. Yeah.
Other questions, comments? Yeah, they're tired, right? Okay, so let's thank Paulo and Bia for their nice presentation.
bensound.com. 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 bensound.com.
Como você achar melhor. Não, não vou usar nada. Não, não. Não, porque eu fiz muita... Pode.
Hello, everyone. I would like to introduce Professor Marco Garcia. Professor Marco Garcia is Associate Professor at the Department of Biophysics and Physiology, Institute of Biomedical Science at Federal University of Juiz de Fora. He has a degree in Physical Education from Federal University of Rio de Janeiro, a degree in Physiotherapy, a master degree, and a PhD in Biomedical Engineer from Federal University of Rio de Janeiro. He developed a postdoc internship in the Department of Physics at USP in area of biomagnetism. Thanks so much, Professor.
Thank you, Lucas, for your kind introduction. And good morning, everyone. Thank you for your presence here. And I'd like to say hi to my wife and daughter who are watching us at YouTube. I love you. So thank you so much for your presence here. Thank you, Bafa, Claudia, Vitor, for inviting me to be here in this exciting workshop. And after being invited to participate in this workshop, of course, I got a little bit nervous. Many prominent scientists here to watching us, and so I guess you enjoy my presentation. So I decided to discuss a subject that I have been dedicated for the last years, the use of surface EMG in motor vocal potential recording. But it's interesting to mention that although it may be canonical in neuroscience, my intention here is to stimulate dialogue on an issue that may contribute to rethinking established perspectives. Therefore, my proposal for obvious reason is not to provide answers, but rather to propose conjectures and critical considerations that may foster reflection on the methodological model commonly employed in map recording. So the title of my presentation is Rethinking Motor Vocal Potentials, the Role of Surface Imaging Shaping TMS Evoked Motor Response. To better comprehend the aim of my presentation, it's necessary to briefly introduce some basic concepts of TMS to those who are not familiar with it. It's well known that TMS allows us to use in two primary modalities, treatment and diagnosis or evaluation. Sorry, no. Both, but how can we explore which one? Only to summarize each modality in the treatment domain, we apply TMS pulse at different intensities and frequencies, which can be organized in sessions separated by minutes, hours, or even days. And in turn, in the diagnosis or evaluation domain, we apply a single TMS pulse, usually set above the resting motor threshold with the aim of recording evoked potentials using electroencephalography, as have been mentioned before in this workshop. It's going to be today also, or tomorrow, by Professor Julio, and or electromyography. So it's essential to note that paired pulse protocols, as have been also mentioned here, presented here by our colleagues, represent a significant advancement in understanding some particular mechanisms underlying the central nervous system function. Since TMS can be used for different purposes, it's usually necessary to determine both the resting or active motor thresholds, as can be seen in this video. Both motor thresholds will help us, for instance, to evaluate the corticospinal stability of a muscle or muscle group in health or disease, as well as to set, it's very important, to set the intensity of stimulation in protocols of treatment using repetitive TMS. It's a very important aspect to consider. However, motor thresholds can be determined subjectively through visual inspection, and a method commonly adopted by clinicians in using electromyography. So, building upon this brief introduction, we now turn our attention more closely to the central subject of my presentation. Interesting to highlight that according to Western colleagues, the resting motor threshold can be overestimated, and it's very important to highlight, by visual inspection or muscle twitch, which is identified in this slide as OMMT. In contrast to using EMG to measure motor cortex response to TMS, visual determination of resting motor threshold in this study was 11.3% higher than when determined using EMG. It appears concerning, considering that it's a modality commonly employed by clinicians in deciding the stimulation intensity and consequently the dosage of repetitive TMS protocols. So, regarding the determination of motor threshold using EMG, as most of us are familiar with, surface electrodes can be placed on the muscle belly, as the slide is shown. And then it's possible to record the motor evoked potential from the myoelectrical activity, as shown in this figure, in this slide. So it's essential to highlight that these issues, as noted by Thierry and colleagues, since in clinical practice, we observe various methodological approaches to determining the motor threshold. According to them, 50% of the studies found in their systematic review used visual or did not provide details of how the motor threshold was determined. The different models, I think, for determining the motor threshold may directly influence the treatment dose and consequently impair treatment and quality, reduce patients' adherence, and increase the risk of adverse effects. We have seen that the approach most often used in clinical practice based on visual inspection may overestimate the motor threshold by at least 10% when compared to using surface EMG. However, the question is surface EMG enough robust for this purpose or for interpreting brain stability and mailing motor cortex mapping? It's in my opinion, is an important question. Recently, Spampinato and colleagues published the present manuscript, and they highlight the complexity of listing action potentials in the cortical spinal pathway and how motor neurons fire, resulting in motor evoked potentials, the parameter that we are interested in. However, it's exciting that they mentioned that effects of dispersion, in this case regarding the map, vary from muscle to muscle depending on the complexity of motor unit innervation, and a very important aspect of this presentation, where some muscles have more than one motor point and conduction distance from the cortex. Curiously, in this study, they present a table, and we have to pay attention to the information that are presented on it, with factors that contribute to map variability and solutions to overcome them. According to this table, the external factors are related to the use of neuronavigation, as have been also very well explained in this workshop, as Victor presented yesterday afternoon. And TMS coil orientation in other aspects of TMS stimulation. But it's interesting to highlight none of them are related to the recommendations for EMG recording. So in my opinion, it's very complicated. We have observed, particularly through the workshop of some research presented at this event, significant advances in the field of TMS, as already mentioned before. That it's reasonable to conclude that these contributions are of measurable value. Neuronavigation, robot-assisted systems provide substantial accuracy and precision in TMS stimulation. However, while substantial progress has been made in optimizing TMS application, it's crucial to revisit the basic principles of map recording, one of which as defined by Groppa and colleagues, which is in case of relaxed target muscle, a positive motor evoked potential response has been defined as a motor evoked potential with 50 microvolts peak-to-peak amplitude. And now, because it's difficult to distinguish motor evoked potentials with a peak-to-peak amplitude or less than 50 microvolts for noise. I don't know your opinion, but according to mine, it doesn't make sense. If you want to make any question or contribute with some information regarding that. I mean, recording a parameter that, of course, depends on the quality of the signal, but to set our metric based on it. I don't know if it's doesn't make sense. So. I don't know, but it does make sense. As most of us and most of our colleagues around the world use this metric to set the motor threshold.
Well, I think an alternative way would be to set this threshold in terms of signal to noise ratio. So if your signal is 10 times higher than the noise, you accept it as a MAP. Because this number can vary from person to person. I think this is what you are.
That's true.
That's what you are trying to say. Right?
Yeah.
I don't know what Risto will comment.
Yeah, one reason it is different in different people is that the electrodes are positioned differently in the muscle, and also the anatomy is different. It can be thick muscle, more tissue between the muscle fibers and the measurement, so it's variable.
But I think it's question that people always look for 50. But it could be 10 if you have a good signal.
For sure.
Or it could be more, I don't know.
It could be more or it could be lower. That's the question. Maybe we should try to move to a concerted action, maybe to redefine the recommendations, because as we have been discussed in this workshop, maybe the way we record and the way we evaluate corticospinal stability sometimes are not maybe the best. Professor, please.
You showed in the camping of resolution, this is to measure the whole curve as a function of intensity, and then in the logarithmic scale, when you take the middle value, that is very reliable, perhaps.
Perhaps. The problem sometimes is how does it it takes, because it sometimes takes too long to do this procedure when you are trying to record the cortical stability. So I have all conjectures, the ideas that I'm going to share with you, and maybe rethink how can we advance on this model, on the way we record motor evoked potentials. I mean, I want to be more provocative to then give some solutions. So please don't. I'm not. Okay. So the idea is we must reflect on it. Our clinicians and researchers looking in the same direction. How far we have truly advanced in the model of recording and interpreting the motor evoked potential. In my opinion, maybe we are stuck, and we have, it's not been very far. Contrasting to, for example, neuronavigation, TMS, robot support, and so on. So I'd like to share some ideas and insights that we have been working on the last few years. So given our concern with the current model and the lack of standardization map recording, we choose to address this matter in our opinion article, aiming to shed light on the issue and to discuss the following question. Can be the recording of motor evoked potential by TMS be optimized? So that was first step on this direction. Among some conjectures and hypotheses presented in the manuscript, we pose the following question. So, does detecting a motor evoked potential from the innervation zone or neuromuscular junction represent a maximization of motor unit actual potential summation in both time and space for the motor units recruited by TMS pools compared with any other specific muscle location. It was an idea. Our study hypothesis that action potential reaching the innervation zone may increase the likelihood of motor unit action potential summation. However, as noted by Saito and colleagues, the spatial distribution of the innervation zone can vary across muscles and individuals. Here we have, for instance, muscles that have been widely adopted in TMS studies, such as the thenar and hypothenar region and the flexor carpi radialis. It's interesting to note how they can be distributed along muscles, no? Different muscles. Even so, the innervation zone still seems to be a suitable target for placing the electrodes. The following animation can be illustrated, as Claudia also used. I had the opportunity to use this very, very nice to present, to use this image. And thank you, Victor, and Andrea for allowing us to use this image. Thank you so much. So you can see a TMS coil placed over M1, and after delivering a TMS pulse, several motor units from a muscle group will be recruited, and motor units' actual potentials will travel along muscle fibers. And map, they will travel along muscle fibers, and a map of amplitude can be determined. However, some variables such as spatial distribution of innervations on muscle fiber directions, type of muscle fibers, muscle compartments recruited by TMS, it is an interesting aspect of this methodological concern. And will contribute to the profile of map spatial distribution. But to better comprehend how surface EMG contributes to map recording, we should discuss the two widely recommended surface electrode montage being applied on TMS, I guess, as I know. One on the left, the one recommended by International Federation of Clinical Neurophysiology, which is based on a pseudo-monopolar montage. And here we have, we highlight that the detection electrode is placed on the muscle belly. And the other, on the right, is based on a bipolar configuration where the electrodes are placed on a muscle location, but it's important to also highlight that it's distant, from the innervation zone. This bipolar montage has been recommended by the CENIAM project, which means surface electromyography for the non-invasive assessment of muscles. Also important to mention that CENIAM is a European concerted action in the biomedical health and research program of the European Union. An important point in the position and protocol suggested by the IFCN is that there seems to be a misconception that muscle belly is hollow, where the IZ is found, which is not always the case. It should be noted that some key reference in the recommendation further corroborate the relationship between the muscle belly and the innervation zone. Here we have an interesting example of how we can be failing to perform the map recording correctly based on IFCN recommendations. It's an atlas. I don't know if you know this atlas. Maybe some know or have the opportunity to use before. An atlas of innervation zones published by Barbarian colleagues that provide the location of neuromuscular junctions for many different muscles. We see that in red for the flexor pollicis brevis muscle, which is widely adopted as reference for mapping recording. The innervation zone is found a little bit distance from the muscle belly, as also reported by Saito and colleagues. Consequently, if we place a single electrode on the muscle belly, I mean we use a pseudomonopolar montage, and according to IFCN, it's possible to record a lower motor evoked potential when compared to that from the innervation zone. I don't know if I'm clear enough, but I mean it's an interesting aspect when we are choosing the location to place our electrodes. In contrast, we have Xenian, as presented here, Xenian recommendations, which suggests a bipolar montage from the muscle belly, but away from the innervation zone. Here we have an example of how the surface electrodes are placed on the biceps brachii muscle, which is based on an anatomical reference, like highlighted here. Following a protocol such as Xenian, so if you use Xenian recommendation, it's an important aspect to be considered. Xenian provides, as previously mentioned, anatomical landmarks which ensure us to reach a greater robustness in map recording. Therefore, it's the image reinforces the need to adopt this reference in case of you use it. So, after presenting overview of IFCN and Xenian recommendation for EMG recording to obtain motor evoked potentials, we can raise some questions. The first, how effective have we been in recording motor evoked potentials using different surface electrode montage? That's, I mean, a good question. The second, is there a more suitable surface electrode montage for map recording? We could be able to set or define new recommendations. It's possible. And the third, the last one, is it possible to determine a new arbitrary value as previous support, a new arbitrary value instead of the 50 microvolts peak-to-peak value based on this new surface electrode montage, or only one, or maybe different montage, which will depend the muscle under analysis. So to simplify the impact of how different electrode montage affect the peak-to-peak value of a motor evoked potential, I would like to share some findings from a study we conducted 5 years ago. We have here a figure exemplifying how electrode montage were adopted in 3 different muscles. I'm going to mention in the next slide. Protocol 1, which meant pseudo-monopolar montage with the active electrode over the innervation zone. And Protocol 2, based on Senior recommendations, which meant 2 surface electrodes over the muscle belly, but away from the innervation zone. We used, sorry, the video turned. It's electronic pen, which is placed over the muscle, and the muscle starts to, we see some twitch. Sorry. Here, it's okay for me, but it's okay. We use electrical stimulation to estimate the innervation zone from the motor point, which is identified in the figure. And the muscle twitch by electrical stimulation provides a suitable with anatomical reference that, in addition to the atlas, as we presented before, allowed us to localize the innervation zone. And 3 muscles, 3 upper limb muscles were investigated from both sides. Biceps brachii, flexor pollicis brevis, and flexor carpi radialis from both the right and the left upper limbs. And according to our preliminary results in the slide, it's also interesting to note that the magnitude of the protocol 1 resulted in 3.5 to 6.1 times higher map peak-to-peak values compared to protocol 2 for the 3 muscles investigated. It's also interesting to note in this figure the variability of each electrode montage. Sure.
So you're discussing which protocol is better, right? Something like that. One is monopolar, the other is bipolar. But at the end of the day, you want to have data so you can compare different situations.
Mm-hmm.
And look to those curves. I mean, if I look the brown one, and if I look the green one, I can probably derive the same conclusions, like the FPB is higher than the others, the FCR is higher than BB, even though let's say the baseline is different.
Mm-hmm.
When you compare it, then relatively, they give you the results. So then looks like you can suppose that I'm working with the protocol 1. You are working in your lab with protocol 2. We can still compare our data and say, well, that's good. I mean, I shoot for FPD. It's better than so. Then I think it's, even though you have, let's say, your input data is different, when you do an analysis, you are always comparing different procedures in the same protocol, and then I think you can derive the same results. So what do you think?
In my opinion, it's a very good question, Bafa. I mean, maybe the solution is to adopt or maybe reach a recommendation regarding normalization of the data instead of using the raw data. And sometimes we see many studies using the raw data. I don't know if it's the best option to use. Maybe if you compare the subject himself, but maybe normalizing the data maybe is the best option. I don't know. But the idea, as I told you, is to compare, because as we see many different studies using different protocols and using the raw data to evaluate corticospinal stability, how can we compare our perspective regarding those data that we are presenting these studies? I don't know. So I have no solution. I want to be only provocative to rethinking about the way we record motor vocal potentials. So another interesting aspect in this data, Renan, Ana Carolina, and I are also studying the effect of each electrode montage on map variability, which can contribute to defining the optimal number of single TMS pulse applied to obtain a robust average value. After that, still sharing some data recorded by, presented by our colleagues. We intend to compare additional surface electrodes on other muscles, like biceps brachii muscle. It's a bigger muscle compared to the other ones. Here we performed the study using IFCN protocol, which meant a pseudomonopolar montage based on muscle belly, as presented this image. A second electrode montage based on adaptation from Senian and Monique and colleagues, which in turn meant a bipolar montage, but with an interelectrode distance greater enough to increase the volume detection, and a third montage based on the protocol one for our from the early study, the study presented before. And according to our findings, it was very interesting, adopting a greater interelectrode distance in a bipolar montage led to higher resting motor thresholds in contrast to the other two montages. It's imperative to highlight that IFCN protocol, it was very interesting, provide motor vocal potentials peak-to-peak values about 2 to 4 times greater than the other two. Well, what should be the next steps to clarify how different electrodes protocols influence map properties and to determine whether standardized framework or muscle-specific approaches are more appropriate? So, multi-channel electromyography represents the most promising approach for established updated recommendations on electrode placement, I guess. And this slide illustrates through a simulation, it's important to mention, how repositioning bipolar montage may influence map peak-to-peak value. While the example on the right displays bipolar recording, let's consider monopolar here only for explanatory purpose. Depending on the electrode positioning, the resulting map, as presented here, may present markedly different peak-to-peak values, as we can see here. So if you have a spatial distribution like we see in this image, and depending on you place your electrodes, maybe we record different peak-to-peak values, motor peak-to-peak values. So, continue on this idea, based on this methodological perspective, we chose to advance the discussion by sharing, by sharing some insights into the application of multi-channel electromyography. So we run a pilot study using a multi-channel EMG to compare surface electrode montage in the brachioradialis into muscles a little bit different from those who are most used, like flexor carpi radialis and so on. So we decided to study brachioradialis and biceps brachii muscles. And we used circular, TMS circular coil. I don't know if the video will be okay. Let's go to try. So it's okay. It's not me. It's much stronger than this guy, I think, and more beautiful. Although he's bald. So we see how can we compare those data. So in the data from the brachioradialis muscle, we see similar motor vocal potentials recorded when extract from the spatial distribution from two different montages, the bipolar and the monopolar. However, when we compare the biceps brachii muscle, I know it's only from one subject, but interesting, because we have two different compartments. The idea of recruiting motor indices is totally different, but if you try to circumscribe an area from those montage, we see how different are the motor vocal potentials. We see here, and the other here, how different they are. So to conclude, I hope I'm on time. We aim to shed light on the methodological aspect that still seems poorly understood in the context of map recording. You see surface EMG signal acquisition. And I think standardized electrode placements protocols are urgently needed to ensure reliable and comparable map recording. In addition, multi-channel EMG holds the greatest promise for reducing variability and guiding robust methodological recommendations. And to finish, new approach to motor thresholds determination may pave the way for innovative metrics and broader applications of TMS. So I would like to finish with this image. I hope we can pave a new way to go on, to move like TMS, robot, neuronavigation have been done. And I'd like to thank you, all my colleagues, NeuroMat, and people that have been contributing a lot to this, my conjectures and ideas, and and I'd like to invite you to maybe to contribute to these speculations and conjectures, because I think motor vocal potential, like TMS, the way we record maybe can be proved. So thank you for taking your time, and obrigado.
Thank much, Professor. Questions?
So thank you for the talk and to summarize many papers and share with us your thoughts. I have a very simple question. I think all those methodological aspects, are important. And in my experience, it's very difficult to make a community agree in how to do some experiment. So I think João is here, but one time we had a network of MRI equipments, and the idea was to collect imaging of diseases that are kind of rare to appear and difficult to obtain a large number, and that could allow a good analysis. And then we had like 3 MRI centers, and everybody was supposed to collect the data in the same way so that we could compare the images. And I'm talking about something very simple. The person will lay in the couch, you push inside the MRI, then you press a button, and then you acquire the image. But at the end, when you collect all the data, you see that something was not done exactly in the center and the other. You have to, I cannot use this data. It was difficult. For instance, sometimes you go there and the person has to acquire the image. You press the button, and the computer say, well, the weight of the person is not appropriate for this kind of the SAR, right? The specific absorption rate is high. What the technician does, he changes the weight, so that's okay, the machine works. But this will pop up at the end. So I think it's very difficult what you're proposing. And perhaps the way is, it's important to be aware of all those issues, but it's important that every group be consistent. I mean, does everything the same way, so you can compare different results. Perhaps, I don't know. So this is my opinion. The other question I want to ask you is that you are measuring electric potentials, and we know that electric potentials, you measure the current that somehow reaches the skin. How this can be affected by, let's say, the fat layer you have between the muscle and the electrodes? Sure. Do you have an experience on that? I mean, if you get a guy or a person that's very thin or a little bit more fat, how this can affect?
For example, senior recommendation, answering the second question first. Regarding the senior recommendations, they suggest how to lead with this kind of variable, how they can influence the result. I mean, Senian suggests recommendations to analyze movement, or they don't have the same, because why maybe it's different, should be different? Because when you are, in my opinion, when you are trying to evaluate corticospinal stability, you are interested on estimating the response when they reach the muscle. So you are evaluating the pathway, not how, if you are, maybe you can also to evaluate how it's spread along the muscle, if you are interested in how different compartments are recruited under a different context. For example, you are trying to comprehend synergism or something like that. I don't know if I'm clear. So what I'm trying to share, maybe we can, for example, use a large electrode over the innervation zone, although a large electrode works low-pass filter, it is a problem, as I have been seeing in some studies. It could be an alternative to recording motor evoked potentials. But it's kind of parameter like anthropometric, fat tissues, also it should be also considered. But if you normalize your data, which should be an alternative too when we record your motor evoked potential, maybe you suppress these problems when you are trying to record your data. And regarding the concern, in relation to the first question, here we have very prominent scientists, and if I say something, maybe I'm not who are Marco Antonio, but who are Risto, Bafa, Claudia. People, of course, would listen to you if you say a different suggestion, because you are reference for us.
I don't think that experience is evidence of being right, so you have to be critical of. And this is the right thing, that you're critical of the established truth. That's really important. But I have this question that, was it so that in one of the papers there was visual observation, so that people just looked at the movement? Is that how they did it, or what was the visual?
The
visual motor threshold determination, what was that? People
it
looking
depends.
At the movement?
At least 10, 11%, but it
Yeah, right, but was it done by a camera automatically, or was it a human being?
Human being.
Okay, or shouldn't that be done automatically with a camera? And the other question is that, how about measuring the force, which is the purpose of the muscle?
Sorry, Professor, I didn't understand.
How about measuring the force?
It could
is
be.
that a reasonable thing? Do people do that?
Sorry, you mean the force? Force? The first. It could be maybe using an accelerometer, or could be alternative to it. Maybe comparing people who have some experience in the clinical practice and compare. It's interesting. We have also done a simple experiment. We asked people who has experience in using visual observation to determine the motor threshold, and in a blind condition, we record the motor evoked potential using EMG, and the difference were very. For example, we record 2,000 microvolts in a motor evoked potential compared to the visual observation. It's totally different. 2,000 microvolts peak-to-peak value.
So I have a question, which in a way is also a response to Bafa's earlier comment about what difference does it make if, or at least maybe that's not such a problem, if when you analyze between muscles, the relative results are the same. And so my question is if that is really what happens. Is there a variability between the many muscles? Because I imagine that for some muscles, the relative position between the innervation zone and muscle belly may be higher or more close together. Mm-hmm. And so maybe these protocols, they end up showing higher differences or lower differences according to the specific anatomy of each muscle. And also, this also suggests that maybe individually, between subjects, the anatomy may change, and then therefore the protocols might not be so consistent across subjects. So my question is if you have any data about this and your experience about this variability.
It's an interesting question, Philippine. According to this atlas, or as I mentioned, maybe if you use high-density surface EMG, we could localize, or using electrostimulation, as presented here, we can suggest using the innervation zone, for example, if it's in the case. But instead of recommending, place your electrode on the muscle belly. And it depends. For example, if you use this muscle, you can see that's a little bit different. If you try to evaluate the corticospinal stability of the biceps brachii, it's a little bit different also. So maybe the recommendation should be try to localize the innervation zone and place your electrode over that spot. Instead, I mean, of using only muscle belly, because it depends on the muscle and how the innervation zone presented, spread over that muscle. And it depends on the compartments. For example, like biceps, you have 2 different compartments, or even some small muscles. But if you place your electrode on the innervation zone, maybe you have a more robust data to evaluate.
We have a time for a quick question and a quick answer.
Okay, sorry.
Because we had lunch. You have a question, right? Maybe I can do it. Just shoot. Shoot and you shoot.
Oh my God. Oh my God.
Respiratory muscles.
Respiratory? I have no experience, but it should be nice. Very nice. But I have no experience. Sorry. Maybe invasive electrodes, wire or something like that. A needle. I have no experience. No experience. I don't know if you have, Claudia. No. But it would be nice. It would be nice.
So this is a topic now for the lunchtime. So we have to adjourn now, and 2 o'clock precisely, we will start with the afternoon session. The session will start with video presentation by Professor Marcel Sibes from São Paulo. He had some agenda issues, so he'll be present remotely, but we can interact with him. So I ask you to be here at 2, and then after his presentation, you have the round table, so we can also discuss all those issues because it's more free time, okay? So thank you again, and see you after lunch.
bensound.com. 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 bensound.com.
Olá, boa tarde.
bensound.com. 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 bensound.com.
Oi, professor Marcelo, você escuta a gente?
Mas tudo bem. So, good afternoon, everyone. We are going to start this afternoon session. And we have a round table, but one of the participants, Professor Marcel Simis, he'll be talking from remotely. So he'll give his presentation first, and then Professor Risto, Claudia, João, and myself will go to the bench, and each one of us will say something about TMS. And after that, the word will be open to everybody. And we want you to engage in this discussion. So the idea is to see TMS, where we are going in this technique, right? Quo vadis. And it's a provocative title. And I hope you can have some thoughts, some questions. And we set the time for less than 5:30. We don't have to reach to 5:30. So if everything is happy, satisfied, we will end earlier. But the idea is to have enough time to promote a discussion. So it's my pleasure now to introduce Professor Marcel Simis. He's a neurologist, neurophysiologist, and physiatric physician. And he's a physician and research at the Albert Einstein Hospital in São Paulo. And he'll be talking about the research he's doing there, okay? So Marcel, the podium is all yours. Go ahead. Thank you.
So Professor Baffa, thank you very much for this invitation and this great opportunity. So as we spoke before, this is going to be a quick presentation so we can open for discussion. And I think the discussion is going to be the most productive part of this talk. I have a few slides just to help, just to guide our talk. So I'm not seeing you, so let me know if you have some problem with my slides, okay? Okay, I assume that you can see my slides, right? And now, can you see the slides showing the Brazilian Society of Neuromodulation?
Yes, you can see.
Yes, okay, so
and,
it's working
we can
good.
Hear loud and clear also.
Okay, very good. So as Professor Baffa was telling me, he spoke before with Professor Maria Elisa Pimentel, and she asked for a presentation that I did during the neurology meeting in June of this year. This meeting was to talk about the Brazilian Society of Neuromodulation, which is a chapter of the International Neuromodulation Society. And in this meeting, we were talking about the clinical use of TMS. So TMS nowadays is a reality in clinical practice, and but there are several challenges of the use of TMS in clinical practice. So here, I think is the great opportunity for us to talk about that. So neuromodulation is not a medical specialty. Neuromodulation is the pool of techniques to use to treat different types of disease and conditions. And is a technique used by several specialties in medicine. So neurosurgery, neurology, psychiatrists, neurophysiologists, physiatrists, anesthesiologists, and others. And especially, so it's important to remember that several health practitioners also use neuromodulation. So it's been used as well for physical therapy, speech therapy, psychologists, nursing, and neuroscience, right, which is very important for the field development. So most of the things that we know and we use in clinical practice came from important studies, such as the one that you've been talking this morning and the previous days. So which been helping a lot, and it's very important for the development of the field. So the idea of the Neuromodulation Society it's a medical society, but the idea is to combine different committees so we can exchange knowledge and improve the field. So this society has some important values, and I would say that the main one is put the patient benefit as the primary goal. If you put the patient benefit as the primary goal, automatically most of the decision also is going to be correct. So you're going to have ethical decisions. So you'll be make statement that not influenced by the conflict of interest. You're going to be using clinical practice based on scientific evidence, and also be consistent with the current law and regulation, right? So about the regulation for physicians, we follow the CFM, that is the medical council, right, for physicians. And so in Brazil is different from some countries in which the different professionals, they follow their own councils, and each council has his own rules, right? So as a physician, we need to follow the CFM. And there are other councils. I think there are people present here in this meeting that can talk better about the other councils than me, right? So I'm going to be talking only about CFM. And so the Council of CFM is from 2012, and it needs to be updated because there is several new evidence of treatment with TMS that's not considered in this resolution from 2012. So from 2012, which is included, it's only TMS to treat depression, and also there is auditory hallucination that is included, the TMS to treat, even though it's not education with high level of evidence. So that it also need to be reviewed to, we suggest that it be excluded, the auditory hallucination in the schizophrenia, so to be more consistent with the literature. And but what are the other indications of TMS that is considered with high level of evidence? So depression, right, is the one that's already included, but also it is considered for most of the meta-analysis and guidelines, chronic pain as well, OCD, nicotine dependence, and in this case with a specific TMS coil to treat nicotine dependence. And there is a situation that's about the upper limb rehabilitation in stroke patients. So there are papers suggesting that it is considered with high level of evidence, but not all the papers is like that. I like the discussion made in this paper, and which the title is also very provocative. The evidence of RTMS for motor and cognitive stroke recovery, hype or hope, right? And I agree with the conclusion of the paper that says that RTMS holds the potential to benefit a range of motor and cognitive outcomes after stroke, but the evidence of efficacy is challenged by the unexplained heterogeneity across many small sample trials. So because of these results that not always consistent, it is difficult to consider TMS stroke rehabilitation as something with high level of evidence, although some patients seem to be benefit from it. We can talk more about this specific topic if you want to after I finish this presentation. And what about other indications that is considered with low level of evidence that, but maybe, but TMS may be used in clinical practice? So fibromyalgia, motor deficit, not only after stroke, but after a spinal cord injury, movement disorders such as Parkinson's disease, aphasia, cognitive impairment, we have a large list of possible application. And what about in Brazil? Is it possible to use TMS for these indications with low level of evidence of efficacy? What we do in clinical practice, we consider this indication as an off-label treatment. And for this special situation, we have this consent form. The patient needs to understand the limitation of the evidence and sign the consent form to agree to receive the treatment, even though it's not recognized by the CFM and is considered with low level of evidence. So what is important here is to have like a very clear and honest communication with the patients, explain the limitation and make the decisions with the patients and the family of the patients. What about the equipment in Brazil? So currently there are some TMS devices that is registered in Anvisa, right? Anvisa is supposed to be similar to FDA in US, but a difference, which is an important difference, is that for some reasons that no invasive brain stimulation devices are considered low risk, and because of that, they don't need to prove the efficacy to be registered in Anvisa. So to have the Anvisa registration doesn't mean it has been clinical tested and approved. So that's also, it's important to have like the medical councils, the other councils guide these regulations, not only by Anvisa, right? Another important topic is the safety. So the risks of TMS technique is well known for indications that has a high level of evidence, such as depression, chronic pain, right? So for depression, TMS is being used around the world and is being approved from FDA for more than 10 years. So the risks is already well known and it's considered safe. But what's important to remember is for several indications, we don't have like large trials, phase 3 or even phase 4 trials. So that is possible. That is still unknown adverse effects of TMS that we will only know in the future. So this is important disclosure that we explain to patients when we are doing this off-label treatment, right? It's something important for the patients to understand. And another important topic is the differentiation of prescription and the administration of the treatment. So we suggest that it has to be well distinguished. What's the professional that can prescribe the treatment and the professional that can administrate the treatment, right? Especially now that some treatment can even be home user treatment, okay? And another important topic is about the neuromodulation in SUS. So it's important to keep in mind that to develop treatment that can be used in SUS, that is the unified health system in Brazil, right? And but to translate these new techniques to SUS is also important to follow the CONITEC criteria to include in SUS, which takes in account the risk benefit of the technique, the cost effectiveness of the technique. So one thing is, if the technique, the efficacy is not well, if the efficacy is not well established, it should not be included in SUS, right? Since we could not do the analysis such as cost effectiveness or risk benefit. So about the presentation, as I said, is a quick presentation. So I think now we can use the opportunity to talk about that and open for discussion.
Okay, Marcel, do you prefer to take questions now or maybe after the others speak? Do you have time to stay with us a little longer?
I can stay. It would be my pleasure to stay with you. But if you want me to answer some questions now,
Yeah,
or
I see if they have
if
some
someone,
burning questions now, so we can ask you.
okay,
If not,
okay.
we move on, and then at the end we can talk. So there is one there.
Thank you, Dr. Marcel. Okay.
Thank you for that. Okay, okay,
Ah,
now I
okay.
can
Go
see you
ahead.
very well. Thank you for coming to talk.
Thank you. I wanted to ask if in your clinical practice, how do you see the patient information, like individual patient information, taking importance on the treatment plan? So, for example, use the MRI data, EEG data to plan the treatment.
Yes, great question.
How do you
So
see that in practice? Thank you.
MRI for sure, right? When we're talking about stroke patients, for instance, the MRI is essential, right? For instance, if you are planning to treat patients with stroke, we need to see the MRI to see where is the lesion, because, for instance, some patient has like a larger lesion, and then we cannot do the stimulation over the lesion, because then we just be stimulating the CFS, not the brain, right? So MRI is essential to planning the treatment. About EEG, I think is important to exclude the patient that has epileptic discharge in EEG. So one of the main risks is to induce epileptic seizures. So one thing that we do, we normally ask for the EEG before the treatment, and patient that has the epileptic discharge, we exclude from TMS treatment. There are new approaches that try to guide based on alpha activity or the frequency of the alpha peak, for instance, but it is still something more related to research than something ready to be used in clinical practice. I don't know if it was clear, my answer.
Yeah, she said it's fine. We have many people sitting in the walls. So I'm telling them to move to the center because I don't know why they are shy. So I think now we perhaps get, oh, there is one more question here. Oh.
Hi, Marcel.
Hi.
Thank you very much for very didactic presentation. And it seems that this is somewhat new society, neuromodulation. For how long? The Brazilian branch.
So the Brazilian neuromodulations, that is Sociedade Brasileira de Neuromodulação, right?
Yeah.
It's not new, right? So, but traditionally, most of the people is neurosurgery, because the International Neuromodulation Society was created by neurosurgeons.
Yeah.
And now we are trying to increase for other medical specialties and even other healthcare professional. So it's been about 3 years that we are doing this movement to try to include other professional in the society. So that's the impression that something new, but it's not.
So in your presentation, you showed that clinical conditions that we have some very good evidence that the treatment is good. and the other ones that we have off-label situations. But these are concerning using TMS as a treatment tool.
Yes.
But we have, especially here, we have many neuroscientists who have been working on as a diagnostic tool. So is there the same you explained for the treatment for as a research tool? Because, for example, in your off-label conditions, you put epilepsy. Epilepsy as a treatment, I think this is pretty much off-label. We don't have many evidence. But as a diagnostic tool, as a non-invasive stimulation, especially in the clinical practice that somewhat we need to see seizures, like in the epilepsy surgery centers, there will be a different situation. So is there a distinction between treatment and as a diagnostic tool?
Perfect. Yes. So there is the conditions, right? That for treatment is not well established. And one thing that's important, we cannot rush much to consider a treatment as something with high level of evidence, because when we consider something as high level of evidence, it creates like a huge impact in society, right? If it's considered something scientific proof to work, right? It's generated several consequences, such as the insurance they have to pay for the treatment, the public service they should include that in the treatment. So that's why it's a huge responsibility to consider something with high level of evidence, right? So that's why sometimes it is better to be more conservative, right? And also to don't generate like a wrong expectation from the patient, right? If you say that scientific proof, so patients they want to receive that treatment, right? They're right to receive, right? They're right to be willing to receive the treatment, right? Okay, but now for diagnosis, one thing that helps to guide the clinical practice is the International Federation of Clinical Neurophysiology. They have a guideline in which they have like the situations they consider it TMS to be validated for diagnosis in clinical practice. If you want to, I can send you this guideline, right? It helps a lot because for diagnosis, right? You know very well that is important to establish the predictive value, right? The false negative, the false positive values, right? To know how accurate is the technique. So there are a few indications in which we consider to be validated for clinical practice, but it's not many.
Okay. Are you hearing us? Okay.
Yes.
Okay, good. I think one burning question also.
Professor, thank you for your presentation. And you mentioned about the guidelines, the recommendations from IFCN. I'd like to hear from you, what's your opinion about using EMG to record the motor evoked potentials? You see people in the clinical ambience using this methodological approach or not? Just for curiosity.
Yes, in clinical practice, you said.
Yeah.
Right. So okay. So I'm sorry if I'm saying something too obvious. Okay, I'm sorry about that. But I don't know how if everyone is familiar with the technique. So for treatment, we have to measure the motor threshold, right? So we do the stimulation in motor cortex, and you see the motor vocal potential. And for clinical practice, we don't need the EMG to establish the motor threshold, because it will not change the parameter that much, right? The knowledge now is considered that it does not change the parameter that much that will influence the treatment, right? Because, for instance, for depression, we have to set the intensity based on motor threshold. Sometimes we use 110%, sometimes we use 120%. So we know that the visual motor threshold is not as precise as the motor threshold measured with EMG, right? But it is not as precise, but it's precise enough for clinical practice, okay, to establish the motor threshold. But this is for treatment, okay? But for diagnosis, for instance, to measure the integrity of the motor cortex pathway, EMG is essential. You need to use that for clinical practice. If you want to just calculate motor threshold to set the parameters to treat depression, is not essential. But if you want to do for diagnosis, that is essential. I don't know if I answer your question.
Yeah, he said yes. So I think we move on now, Marcel, and I will compose the panel.
Very good.
I'd like to start to call Professor Claudia Vargas from Federal University of Rio de Janeiro, Professor Risto Iumoniemi from Aalto University in Finland, and Professor João Leite from the Ribeirão Preto USP Medical School to sit here, and myself. So please, you sit in the center.
Yes, I'm here. I can hear you. Just leave the do you want me to leave the camera on, or can you leave off?
Não está compartilhando? Mas ele pode ver. Marcel, alô, Marcel, você consegue ver através do...
We can see up.
Tá excelente, eu estou vendo vocês na mesa.
Está vendo na mesa, mas não está vendo a transparência?
Não.
Talvez, se você pudesse assistir pelo YouTube, que aí...
Perfeito, vou fazer isso.
Tá
Vou
bom.
fechar aqui o microfone para não entrar e eu assisto pelo YouTube. Perfeito. Mas aí acho que a ideia do YouTube funciona bem.
O
realmente.
YouTube é melhor, você pode... no YouTube você vai ver tudo, e aí...
Não, é só compartilhar.
Compartilha ali. Não, eu não tenho como... Quem está na ligação? Oi?
Se for muito importante ele ver a apresentação, você vai ter que entrar no Google Meet com um convidado para que você possa compartilhar aqui.
Mas aí não fica tudo mais enrolado.
Vai demorar um pouquinho mais, vai ter que entrar na sua conta aqui.
Não, acho que, Marcelo...
Eu testei pelo YouTube, tem um problema YouTube, tem um delay grande, de uns 5 segundos. Aí para discussão acho que vai ficar travada. Mas
Se você quiser apresentar...
Mas eu acho para discussão, né? Mas para apresentação eu assisto por lá, então.
Cadê a apresentação?
Pega a apresentação e põe aqui, ó. O seu e-mail. Técnico.
of give this very provocative name, so where you are going, TMS, right? And this year, Transcranial Magnetic Stimulation is celebrating 4 years of existence since the first time that Tony Barker, Friesman, and Hanolius demonstrate the technique in London. So I think it's a good time to think about the future. And to think about the future, we also must think about the past and the present. So my introduction will be somehow to show you a very personal view of this area and how I got into that and how probably other people in the bench also got involved with TMS. Okay, so we know that the principles of producing magnetic stimulation in the brain are actually a form of producing electric fields in the brain. And to produce electric fields in the brain or currents, we need to apply a time-varying magnetic field, like those Maxwell's equations show. And if you have a very high rate of changing magnetic field in time, you are going to have a very strong electric field, and this could produce currents, and currents at the end can depolarize neurons, right? So there is some history associated with TMS. I think the first one that thought they were seeing transcranial magnetic stimulation or brain stimulation was Dunserval in 1886. And actually, he was seeing not brain stimulation, but what we call phosphines. So phosphines are depolarization of the retinal cells. So if you close your eyes, relax, and apply a gentle pressure in the eye, you start to see flashes, right? So this is what we call phosphines. And then put my glass here. Then in 1959, there were some magnetic stimulations of the frog sciatic nerve, more or less like Galvani used to do in the early times of getting a frog exposed to a thunderstorm, and then the frog will move the legs. And then in 1965, there was this proof of concept by Bickford and Fleming showed that they could stimulate the peripheral nerve. And finally, in 1985, Tony Barker and his team showed the stimulation of the brain. So just a picture of the first device that was built by Dunserval. You can see it was a big coil, and they were applying let's see the pointer here. So Dunserval is the guy that's at the right, left. Yeah, I don't know, at the right of the picture. At the right of the picture is the guy with a big mustache. You can see that the head of the lab, you can see who they are by the size of the mustache. So that's Dunserval. All right, and this is another interesting thing. You know, physicists working in nuclear physics, they built this very big machine that needs magnetic field. And they also reported that this is a cyclotron. You can see between those two flat, in this gap, we have a very strong magnetic field. You can accelerate particles there. So this is today a tool used in several areas in medical physics to produce radioisotopes and even proton therapy. Now some machines use the cyclotrons. And you can see Lawrence there. And people will report that when they shake the head near the magnetic poles, they will have phosphines also producing their eye. And this is another picture now of transcranial magnetic stimulation now by Tony Barker. You can see in the left of the picture, and in the right you can see his collaborator,
Reza Janolius, Ian Freestone, and then Tony Barker. I first had my transcranial magnetic stimulation was in a conference, the World Congress of Medical Physics and Bioengineering in Kyoto, and Tony Barker was there stimulating people, and everybody got very excited. Okay, another important landmark, I think, was produced by Shogo Ueno, as Risto showed in the first presentation. Tony Barker and his team use a circular coil, and the circular coil will produce a stimulation that's kind of spread in the brain. But Shogo Ueno had this idea of using two coils, and I just got one here from MagVenturi stand, and you can take a look how they are. So they are two coils that they are wrapped, and at the center, they are superimposed, and they somehow add the current, so you can increase the field intensity you produce, and you can also increase the focality, so you have a more restricted region. So this also expanded by significantly the field, and now we have many coil designs. Probably Professor Risto, you will talk about it. But Shogo Ueno came from the biomagnetism community, and people working in biomagnetism, they struggle to get good signal. And we have to produce their gradiometers. And if you look to this 8-shape coil, it is very similar to what we call a planar gradiometer. This thing you see here, you have two circles and they are connected. So if the separation of the coils, we call the baseline. So if you make the baseline 0, then you get a figure of 8 coil. So it's the same design. So perhaps Shogo was somehow stimulated or motivated by the kind of work we doing in biomagnetism. So here I have another picture. How I make the laser point work here. Como é que eu faço aqui? Não, mas como? Tem que sair. Okay, good. Yeah. Every time you are lost in informatics, you call a student, and then the problem is solved. So here you can see a picture of the BioMag steering committee. You know, it's a group of people that organize the biomagnetism conference. And here you can see a guy named by David Coyne. David Coyne was a senior member of this committee. He was one of the founding fathers. And when he learned about TMS, he was really excited and brought this subject to the community, always advertising it, that could be something interesting and telling all the good benefits of a TMS. And here you can see Shogo Ueno. And here you can see Risto Umoniemi, this picture. And right there in the back, you can see also Gianluca Romani from Chieti University now, and he's also working TMS. So as you can see, TMS was invented in England. Many groups were using it, and there was this community that was studying magnetic fields in the body, but not applying magnetic fields, but instead measuring the magnetic fields that's produced by the body. So exactly the opposite. And these are people that had and still are having a good influence on the field. So I'm sure, Risto, you talk more about other developments. Yeah. Okay, that's a good point. All right, so this is a big, the whole picture here. Of course, we started also working in TMS in Brazil. And there are many groups now. I don't want to be exhaustive, but so the NeuroMat project has a group, and in this picture you can see pretty much everybody that's involved in our research. And here is also Professor Gauss, the founding of the project, that I want to also thank him for all the support. So now we have to think about how about the future, right? What's TMS is promising? So we are having a very intense agenda here. So yesterday I did not have too much time. So I asked my artificial intelligence machine and said, what's the future of transcranial magnetic stimulation? So that's what I got as a response. The future of transcranial magnetic stimulation involves more precise targeting via AI and neuronavigation. So we talk a lot about neuronavigation here. So the machine is somehow 50% right. Accelerate treatment protocols and personalized medicine using genetics and neuroimaging data, and expansion beyond the clinic with portable at-home devices for broader accessibility. Somehow we said things around this area. And research is also expanding into new applications, such as eating disorders and post-stroke cognitive issues, by focusing on network level changes and multimodal imaging to understand mechanisms and boost neuroplasticity. Okay. Okay, you should give a negative, no, thumbs down, right? Okay, how about the technological and treatment advancement? So they are talking about precision and AI-driven protocols. AI and neuronavigation will enable hyper-precise targeting of brain networks for optimized therapy and reduce side effects. To accelerate and personalize treatments, clinically intensive regimes will condense treatment times. We know how difficult sometimes it is to the patient to stick with the treatment, and personalized medicine approach will combine genetic and data and neuroimaging for a more, let's say, subject-specific therapy. And at home care, so this is very important. We know the population is aging. Probably you won't have hospital to everybody in a few years. So then you have to do some treatment at home, and then portable devices could be an exit for this problem. And also this hybrid clinical models. So we can go on, and there is now applications. So probably we'll see something like by João, I hope. And TMS is being explored for showing promising results for conditions like eating disorder, like anorexia nervosa or bulimia nervosa, and post-stroke cognitive impairments. And multimodal research combining TMS, EEG, and MRI, EMG also, right? Should write here, okay? And it's crucial for understand the effects of TMS on neuroplasticity and network level changes. And finally, the key trends are understanding, boosting neuroplasticity through modality research in key to improve treatment efficacy and long-term outcomes. And network level focus future research will shift from single brain region to analyze network level change to provide a broader insight into the effects of TMS. I think this is exactly what the multi-logs TMS intends to do, probing network. And ethical consideration is also a big problem. I think Marcel touched on that. The growth of a TMS technology necessitates a holistic analysis of ethical considerations alongside practical challenges for successful implementation. So Marcel mentioned, at the beginning, the issue of safety. And most of the equipment we have developed up to this moment was very restricted on this issue. I mean, we want to benefit the patient and stimulate the brain and nothing else. But in the recent years, because many patients with psychiatry disorders need to go to an electrical convulsive therapy, people are now, let's say, releasing all the breaks we have in a TMS machine, so we can produce a magnetoconvulsive therapy. And in this case, it seems to be better than electrically convulsive therapy. So this is something that opens a completely new horizon for the technique. And I think the people in the panel has a lot to contribute. So are you curious to see more? So this is curiosity in Mars, still walk around and collecting information there. And so I stop here, and I was thinking starting with Risto, technological aspects, or another, and then Claudia, and then João. I must say that I didn't put the picture of Risto after Shogos, because he's here present, so I don't need to show his picture, but Risto is a person that has done many improvements in the TMS area. He probably you saw his talk in the first day, coil design, stimulator design, mapping and everything. So he can name it and you talk to him and he knows, and he has contributed to it. So I would like to stop now and turn the word to Risto, and I will stop my presentation so we can go to the normal mode, I guess. I think now, Marcelo, can you show, Marcelo, pode mostrar Marcelo, você consegue nos ouvir?
Sim, estou ouvindo super bem.
Mas
estou ouvindo bem.
Gente, não, né?
Oi?
Está vendo, não?
Eu estou vendo a mesa, estou vendo bem a mesa.
Está vendo a mesa? Ok, então tá bom. So, Risto, we can start. Marcel is seeing all of us, so you have the word now.
Okay, thanks for your nice presentation. And asking AI what the future will be like. So I was thinking that what will the AI tell in the future? So it will include EMG. And but it will also include other things like combining TMS with other kinds of stimulation. So the most easiest way to stimulate is sound, like music, and vision, touch, the already stimulators like that, and combining it with TMS. And also, like say in stroke therapy, maybe in the future, people more and more do the TMS and practice with your limbs at the same time. And also, I think there's a big potential in combining TMS with pharmaceuticals. There are some attempts now to investigate this with psychedelics and with the other drugs as well. I'm not a this is not my area of expertise, but I think that's going to happen. And automation, it was mentioned that it will be faster in the future. There's a lot to do there. Maybe even kiosks where you can go and press a button and so on. Maybe two other things. Antonio gave this talk on theory of the neural networks, and I think in the future we will learn more about how the brain works, And of course, then there's a lot of experimental studies and models of how the brain works to have less trial and error research. So nowadays it's quite a lot. Like that, you try something and without full understanding. Of course, I don't know when we're going to have full understanding of the brain, but we should aim at understanding the brain better. So we need everything in neuroscience and theory also in this. Well, maybe that's enough from me.
Claudia?
And it would be really nice to discuss with everybody here.
Yeah. Thank you, Liz. Yes. Please ask questions and make comments, right? Yes, so the image that comes to me is that this new technology, as Bafa put very well in a very nice presentation, opens up a sea of ignorance, right? Where we are faced with a number of new questions and new possibilities that come with new technologies, and also what to do with the available technologies. So in this morning, in my presentation, I was trying to figure out how to put together data coming from different technical approaches to answer the same question, more or less the same question. So we are still on the verge of these combinations, and of course this will be even more challenging as we move towards new technological approaches. So I like very much the idea of combining TMS with paradigms where we challenge the brain with different sensory modalities. I think this is a new field, and I mean there are so many open fields to come, and I can't wait to see how this. So that's for now.
First of all, I would like to thank Bafa. He invited me, in fact, Sunday night. Let's see, João, you want to participate in this roundtable? And I think that I'm very glad for that. And I'm going to tell a story about Bafa. And when we joined the project, and it was more than 15 years ago, FAPESP was trying to invest in very good equipment. And at that time, we didn't have many epilepsy surgery centers, or they are lacking for very good equipment. And at that time, we are discussing with other colleagues, neurologists, basic scientists, and BAFA, and we were imagining, it was exactly like we are now, thinking that what would we have epilepsy gaining a very superior level in 10, 15 years. But FAPESP didn't have much money, and we have to invest in state-of-the-art equipment at that time. And I remember when we were trying to use that money, of course, it was a few centers, 4 or 5 centers in the state of São Paulo. And BAFA brought that TMS would be, in fact, magnetic MEG, and it was a very expensive equipment at that time. It's still, nowadays, it's very expensive. And then we are discussing things, and then at that time, we were lacking MRI machines. And then, so we decided to give the priority for those equipment. And with that situation, we created a program in FAPESP, was called Synapse. And with that program, we were able to develop very good epilepsy surgery centers with imaging. So here, Campinas, São Paulo, and BAFA joined us and in fact was one of the mentors of this project, even without getting his state-of-the-art equipment. And I'm very glad that in recent months, I think, we started talk about using this very brand new neuromodulation equipment in clinical scenario, especially looking for epilepsy treatment. And I know that Marcel mentioned that this is epilepsy is not a it's not a confirmed or one of the diseases that are that it's already proved that it will be used. But we don't see, and I think that we don't see neuromodulation, at least it's my opinion. I don't see neuromodulation as a powerful tool to treat epilepsy. In fact, these are many technical and methodological problems. And we are experiencing neuromodulation approaches in epilepsy, but they are only, I would say, palliative. We have a very good neurologist who has been working with depth electrodes, recording patients. And when we suggest neuromodulation for those patients, it's because we could not find the epileptogenic zone. So for now, if you go to all centers, we did the gold standard is to find an area that we could remove it, and without giving that it, of course. And then we can say many times that we can cure patients. The other situations, it's really, really hard, and we can improve it, but it's much more limited. But I think that this is a very unexplored approach to use neuromodulation in TMS, and especially with this very elegant equipment that BAFA just bought, and to use it as a non-invasive approach to dissect the circuits that would be, we believe that they are participants of this epileptogenic zone. So this, together with EEG, because we need to have a functional idea, and with MRI, structural MRI, and fMRI, I think that will be a very good scenario to treat patients, especially the ones that are most challenging ones, that the ones that we don't see lesions. Because MRI, and I think that our choice 15 years ago for MRI, I think was correct, especially at that time that imaging was a boost in neurology field, but especially in the epileptology. But now we are facing situations that we don't see. Many times the brain looks normal, or we don't see structural lesions, but the patient has intractable seizures, have tried many different anti-seizure medication. So I believe that this is going to be a very good situation to use it as a diagnostic to dissect. So that's at least my dream to use, and it's especially to convince the young ones, the very smart guys, to will bring the problems, and maybe you can bring us solutions for these patients. Thank you.
Okay, João, thank you. So the word is open now to discussions. And I see here, okay, one in the back, but just I see here Professor Adilton from our department. He's my chair, and he used to work in biomagnetism, and now he's working with ultrasound. So I want to ask you, Adilton and Risto, because both of you touched this question of a multimodal stimulation. How do you see the possibility of using high-intensity focused ultrasound and TMS at the same time? Is it possible? Would it be any gain, or has anyone done that? I don't know.
Yeah, thank you, Professor Baffa. Thank you for introduction. This is my first question. I'd like to contribute with this discussion with Professor Rizzo. Yeah, there is many paper, many applications we use factor ultrasound because best resolution in the brain target, for example. And I don't follow well recently, but there is some application use ultrasound and MRI and TMS together because you can coupling both. And considering some results, it's so effective. But I'd like to listen, maybe it was a question for myself about the safety. Okay, but anyway, the big problem of ultrasound is the scalp, is the bone. I remember 5 years ago, I was a PhD student, and the main project of this PhD student was look for the best protocol of ultrasound to brain stimulation, because when you use just AC stimulation, for example, just a pulse, you have a reverberation inside of the brain because the scalp. And it's create a small, I forgot this, small blooding inside of the brain. It's a general, because that ultrasound for brain stimulation was stopped. But after that new protocol of stimulation, where to avoid concentration of waves inside of the brain, it's start again a high application. Recently in São Paulo, in the I think Albert Einstein Hospital, was installed a system, hybrid system with together with MRI, and it's increased a lot. But unfortunately, I don't follow well this protocol, how safety is for ultrasound, for example, compared with the TMS. I have a doubt about this. If some guy from the table could help or some news about this, I would like to listen. But anyway, as Professor Baffa told, it's very, very interesting and very useful to be together. Okay, it's possible. Two different energies, physical protocols, ultrasound waves and electromagnetic waves. So interesting. Thank you.
You want to comment or?
Well, you're asking about safety, right? Of ultrasound. I don't really know, but of course it is really an interesting technique if you want use it to ablate in tumor surgery or maybe even epilepsy to cut some connections. But I guess you're asking about the neuromodulation use of ultrasound. Yeah, I don't have any knowledge of how safe it is. So,
yeah, I think Marcel wants to comment. I don't know. Go ahead, Marcel.
So, Professor Baffa, about the ultrasound, right? When we talk about the ultrasound, we are talking about a broad spectrum, right, of ultrasound, right? So, ultrasound is being used a lot in clinical practice for transcranial ultrasound for diagnosis, right? We do like transcranial Doppler analysis with ultrasound, right? And it's safe because the intensity, right? And about the combination, there is a group that did a study combine TDCS, that is electrical current, with ultrasound, and with the hypothesis that the ultrasound helps to guide the electric field and makes it goes deeper in the brain and to reach basal ganglions in the brain. So there is some people trying to combine ultrasound with TDCS. I don't know so far with TMS, right? And then there is studies with ultrasound that is with single pulse ultrasound. There is some studies with Alzheimer's disease, and the hypothesis is that this pulsive, this ultrasound pulsatile, just pulse of ultrasound can help the brain if patients with Alzheimer. But the mechanism is a little bit is different from TDCS and TMS, because you are probably acting in the glymphatic system, so you helping the brain to reabsorb the metabolic in the brain. So it's a different approach, right? And the system that we are using in Albert Einstein Hospital is called HIFU, high intensity focal ultrasound. That is an ultrasound combined with MRI, and in which we can do like a very, very small lesion in the brain with a millimetric diameter, and it's very precise. So it's a way to do like a lesion in the brain, and it's already approved to treat a tremor in Parkinson's disease and tremor in the essential tremor disease. But I don't know if I answer everything, so if it's clear my answer.
Yeah, I'd like to give a complementary information, Marcelo. Thank you. As a Professor Issa told, ultrasound can use both for neuromodulation and as a therapy for neurostimulation. As you told, in the Albert Einstein is for therapy for stimulation for heating. You create a lesion with a high precise focused ultrasound. In this case, is a treatment. But when on the past, new past, like 5 years ago, you mind that you need a high-intensity ultrasound for neuromodulation. But recently, you described that it's not necessarily high intensity. So in this case, it's not. It's safety. It's not have a problem. Just as neuromodulation, because you're not ultra poration, for example, you're not opening the channel by ultrasound first as you might before. Just some target, mechanical target inside of the brain, and it creates a drug delivery, for example. So as a combined ultrasound with some drugs, it's so useful for Alzheimer's, for example, treatment and any others. In this case, it's not so stimulated, just neuromodulation. It's low intensity focused ultrasound. So it's very, very interesting in this case. And together with TMS, this low intensity ultrasound for just neuromodulation, together with some stimulation for diagnostic, as told Professor João, for example. It's so interesting for drug delivery and for diagnostic.
Okay, thank you. I think there is another question. And as he brings the microphone there, you work in ultrasound, I work in TMS. So let's band together and try to do something. Okay. Yeah, it's só falar que.
Hello, everyone. I'm a child neurologist at the same department of Dr. João, and I see the TMS as a promising therapy. So my question is about what is the opinion of you about the potential limitations on TMS application in children? The size of the head, the height, the required of cooperation. It is possible sedation, or it requires anesthesia, something about, I think that the profile of disorders in child neurology, it could be promising TMS on some kind of disorder. So I realized that in the future, as AI didn't talk, the children could be a potential population. So I want to hear you about the limitations of future on children.
I think I have on the table people more informed about that, but I think you touch a very important issue. We know, just in my opinion, just little about child development. and how brain stimulation can steer this child development. We see now many kids that are in the autism spectrum, and we don't know how to do. And now that I learn about that, I look around and I see many friends that probably when I was a student will be in this class. But anyhow, joking apart, I think we have several challenges. So coil design is one of them. The first equipment, they have like large circular coils for adults and smaller circular coils for the child. I think now MagVenture has on the booth there many small coils that could be suitable for child. You touch another issue, that's cooperation. I know we run experiments in our lab that even ourselves sometimes got tired because it takes 1 hour, 2 hours until you prepare everybody, set electrodes, put the EEG cap, check for impedance values, and then go there, scratch again, and put some gels. So child won't do that. I mean, that's forget about. So then I think you, from the technological or from the medical physics point of view, we have a lot to do, right? And I don't know about the other issues. Perhaps Marcel can comment, or Risto, I don't know. Have you done any child measurement in Finland, or?
Not that I know. It doesn't look very dangerous, but I think there are ethical requirements that you do not do scientific studies in children unless it's the only way. And of course, if there's need for surgery and you want to find where the motor cortex is, that would balance the risk, if there's any risk. An interesting thing is that the smaller the head, the stronger, because the electric currents are confined to a smaller space. On the other hand, you may be able to put the coil closer to the brain because the skull of the child is thinner, so they probably compensate. Child studies have been made. There were papers a long time ago on the safety, and it was considered safe in published literature. Also, EEG, it's a long time ago when I read, but EEG was measured. There's still discussion that why are the TMS EEG responses much bigger in children than in adults. Yeah, well, that's all I know about. That's not my area.
Marcel, you want to comment?
This is a great question because children we have a especially concern about it. The major concern is about the safe, right, of the neuromodulation in children. The major concern is about what's the interaction of the neuromodulation with the brain development. So we don't know, we are changing the way the brain are developing, right? So in some situations, it seems that can be justified. For instance, in cerebral palsies, right? We know there is a lesion. We know that this brain will develop with a natural we know that this brain will develop with maladaptive changes related to the lesion in cerebral palsies. So may justify to try to treat these maladaptive changes with TMS, right? So for some situations, it's better no, and it seems to be justified. For instance, cerebral palsies. Although for autism and ADHD, TDAH, right? We don't know much. We don't have much data about the benefit. We don't have much data about the safety as well. That's a concern. We have some papers, meta-analysis, showing that it's safe so far. But what we know is from these exploratory studies with a small sample size and a small and a short time of follow-up, we don't know what is the impact in long term. So me myself personally, I don't use neuromodulation in clinical practice because of these reasons. What we are doing, we are developing trials, so we may see how is the response of the children in clinical trials, but not in as off-label treatment. Okay, I don't know if it was clear my answer.
Just addressing this, I think that in epileptology, we are facing a similar situation as in cerebral palsy, when we have, especially in children with catastrophic epilepsy, which we have to do something to, especially the ones that have many seizures, and this compromises the normal development. So in this situation, I think it will be justifiable. You have to remember that when we are TMS, it's much less than invasive than putting depth electrodes and stimulating people, or in a telemetry unit, we use we move the antiepileptic medicines that we want to see patients seizing. So it's a safe situation because we have all the nurses and doctors. And so in this situation, I think that an attempt to use this as to stop seizures would be justifiable as in cerebral palsy. I think that then in these cases, we see the dramatic improvement when we, for instance, remove the epileptogenic lesion, and some children start to catch up and start to learn, and especially in the catastrophic, we have an entity that is we call catastrophic epileptic situation, that sometimes the children, they have like one hemisphere sick, like with the cortical dysplasia and other disease. So this it's a situation that probably it's justifiable to try to improve it.
Okay, we have one question by Duby.
It's not a question, it's more a comment. I agree with Professor Marcel that the main concern in children and stimulating kids is the actually neuronal development, whereas you in the same situation you have a little bit contradictory information or practices. On December 22nd, 2022, the neuromodulation in Europe was classified or reclassified as a class 3 medical devices, so the highest risk. And exactly for this reason of uncertain effect on neuronal development. Now, very recently, like weeks ago, in US, you have FDA-approved protocols to stimulate adolescents for depression, right? So the line of neuronal development is very blurred, okay? So and also we are talking about two regions, so Europe and US. So US doesn't have it as a class 3, it has it as a class 2 device. But I mean, international consensus, scientific consensus of whether it's safe or not is very diverse, depending on the region, and the interests actually are completely different, So there is no unanimous scientific evidence or consensus that it is safe or it's not safe. Just to comment that.
And how do you think this could be overcome? Any hint? I mean, what kind of studies people can do to perhaps, because I understand from the clinical point of view,
you are kind of desperate trying to help that child. But so you think research with animal model in different stages of development could help? What would be the suggestion for people that are here or listen to us?
I mean, it's do more research, but that's the immediate recommendation. The thing is that, this kind of research on effects of TMS on long-term neuronal development is notoriously hard to set up, if maybe even impossible. I think the longitudinal studies are one of the solutions to really monitor everyone who was in some kind of adolescent age, a TMS subject or had a treatment in TMS. That would be one of the solutions. But definitely, research, more research in this area.
Thank you. I think Risto wants to comment.
I just want to clarify what is meant by safety in children or also adults. There are two things. One is that if you give a pulse, is there some mechanical, there could in principle be heating, no danger like that in adults or in children. Is there some mechanical danger? No. But the risk is, and I think you already pointed out that it is really if you do treatment like let's say depression therapy where you give 60,000 or 90,000 pulses or something like that, maybe it has some adverse effects. So it's a different thing. It's in the literature, it's considered safe if you just give a small number of pulses, no damage. And so it's an ethical thing if you try to, let's say, improve ADHD or autism or just a healthy person to try to make memory better or whatever. So there are risks, and even if there wouldn't be risks, it's still a major ethical question. Is it okay to improve people with technology somehow? Of course, sick people, if there's epilepsy or tumor, that has to be treated. But
okay, I think there is one question there in the back.
Yeah.
Oh, here.
Well, was faster than me. I was going to mention that exactly, that in the US a few weeks ago was approved for adolescents. So yeah.
Hello, Professor Marcel and everyone in the table. Nowadays, I have a lot of students visiting the university, especially now that the robotics and the neuronavigation is in the news, and we have television visiting. And we try to explain how it works and the main brain targets that are used, for instance, to treat depression and schizophrenia, hallucinations, and everything. Can you tell us a little bit more about the main brain targets that are used to treat, for instance, tinnitus or hallucinations? Because in the depression, we know already that the left dorsolateral prefrontal cortex, especially in relation to EEG electrodes, so the F3 electrode is very used as a kind of reference outside the head. But I'd like to know your experience about other important brain targets for these other diseases.
So for tinnitus, the target is to do the inhibition of the auditory cortex bilateral, right? Since the auditory is processing bilateral, you do the inhibition bilateral, which is in temporal lobe, right? Superior gyrus of temporal lobe. That is T7 and T8 bilateral. You do this 1 hertz pulses inhibiting that. The literature is suggesting that the better results is when you do the priming of with the stimulation of dorsolateral prefrontal, the same area that you do to treatment of depression, right? So the protocol normally you prime with the stimulation of dorsolateral prefrontal, and then you do a bilateral stimulation with TMS. And but the results for tinnitus is very heterogeneous as well. So some patients respond very well, others do not. Most of the patients do not respond. And some of the patients that respond, the tinnitus can come back after 1 month, 2 months later. So sometimes it not considered something that is clinical significant, right? That's another important concept, because sometimes it's statistical significant, but sometimes it's not a clinical significant, right? Okay, was that the question? Thank you.
So more thoughts? Thais?
I would like to ask, in your opinion, how TMS was used as a tool to understand neurophysiology? Like, what, in your opinion, was the major contributions for TMS to understand circuit communication, neuroactivation, and so on?
Well, you do experiments. For example, double pulse stimulation, and how does the first stimulus influence the, let's say, motor response of the second pulse? You have the data, and people have been thinking about trying to explain what happens, and one can also include pharmaceuticals that influence, for example, glutaminergic transmission, and they can then understand something about the processes there. So it is like combining experimentation and trying to understand what is going on with different kinds of manipulations. That's maybe somebody else will compliment.
Well, this morning I gave some examples of how TMS can be used to map plasticity in the sensory motor cortex. I think it's pretty well established, although there is some discussion in the literature regarding other methods as I brought, but I think it's pretty much established. So it's another bunch of evidence that has been brought forth by TMS. And of course, all this literature that Professor Unomiemi just mentioned on the homeostatic plasticity and all the mechanisms that go that come with these changes in the brain induced by paired pulses and repetitive pulse. So there is a large bunch of literature on this. So I think this is a very important contribution of the classical TMS. Let's put like this, because as I said in the beginning, I think that we are going to see a lot more in the forthcoming years.
Okay.
Yeah, I ask this because the new technology comes, and then we see and we analyze data, and we see, okay, maybe this also has an impact on MEPs or EEG. So it's a building process. So yeah, thank you. I guess Professor Histo said something interesting about the paradigm that sometimes is used, like we get the data and then we start coupling more knowledge to try to explain what we are actually seeing. And I guess that has some impact on the clinical side, because it's also kind of experimental practices.
I'd like to comment some things. One time I went to the library and saw this big book that was a facsimile of all Leonardo da Vinci's anatomic studies. And so I was flipping the pages, and then you can see the skeleton very well drawn and all the things very precise. And then you turn the page to the heart, the circulatory system. Perfect. You can see all the chambers, the arteries and veins. But then you flip the page for the brain. And then you could see a good drawing of the eye, a good drawing of the optical nerve, and then a big mass, completely homogeneous. So that's what he knew in that time. Okay, then medicine, progress. You have anesthesia, you have surgery, you have X-ray came. So 1895. So it's not 1985, it's 1895. Somehow close to the TMS. And then, Röntgen found this mysterious ray that he called X. And this could see the interior of the body. And boy, this gained a widespread attention. People were X-raying women to see what was underneath the skirts. And they were using X-ray for everything. And so this opened a new window for medicine because you could see inside the body, at least the hard tissues, without having to open it. Okay, then we have some more time. And then in the 70s, like just after Second World War, people were playing with ultrasound also. They got a tank turret, the thing that goes around and shoots, and they put a person there in a water tank, and they were able to make some ultrasound images because sonar was being developed for the submarine. So the technology opened this window. But then in the 70s, we have X-ray tomography. So now we could make slice. We don't have a projection like Risto showed in his first class, put a glass and you have just a projection of one direction. So you could have a set of slices so you can see they used to call TAC, right? It's axial computation. I forgot the name now. CAT, right? Computer assisted. So anyhow, you could see the internal part of the body. And then, MRI came, and also this opened another completely different window. And we have all those magnetic techniques like MEG and TMS now that are kind of complementary, right? Because one measures the fields that are produced by the neurons, and the other excite the neurons by magnetic field. And what's good about magnetic fields is that our body is transparent to magnetic field. So if you try to shine light from one side to the other, the light will be scattered, it doesn't go through. But magnetic field pass through. So I think what we are seeing now is the opening of a new window to see how brain works. I think it's 40 years appears a long time, but not for science and for technological development. When you see the first equipments of TMS, they had the first ones had even valves, vacuum tube valves, tyrotrons to connect the circuit and disconnect it. And then we have a lot of evolution now, and you saw the equipment we have. They are small. They can deliver a lot of energy. So we are opening windows, and this is one more window we are opening. And so I have seen things that, in Risto's lab, they were, inducing aphasia in a patient. So you can see an object and name it, and then all of a sudden you zap the area there associated with the speech, and the person won't speak anymore, won't be able to name what he or she is seeing. So I think there is this many new possibilities, and of course we have let's say our methods we have much more means, but let's remember we are not dealing with a simple object. The brain is not a simple thing. That's what makes us to be here, and I think there are complexity, everything. So That's why we have to keep moving, right? We have to keep pushing the area forward, improving the technology. We see what happened to our lab. I mean, when we started with André, he's there, we said, well, we are physicists, let's build one TMS machine. And then, okay, let's see, make some drawings, go into the literature. And then I said, well, let's order the parts. So I went to there was no internet at that time. So I went to call people and ask for prices and then say, oh, this is too expensive. And then I saw a Russian equipment, very simple, very robust, that was selling by about the same price of the parts. Okay, let's buy it. We bought it. And then let's do a motor mapping. So what we did at that time was to put a swimming cap on the person's head, draw some Cartesian system of coordinates here, and let's put the coil in every point and measure the map. And after a few trials, we said, well, there is some problems here. We don't have accuracy, right? We don't have precision, not accuracy, precision. Because you could do the person several times and the results would be different. And then you realize that, the positioning was not good. I mean, the X and Y and Z. And also the angle. I mean, the angle is very important. And why it's very important? It's because magnetic fields are not monopolar. They are dipolar. So the lines always wanted to go from the North Pole to the South Pole. So they spread. So you cannot focus like a wave in ultrasound or, I don't know, electric field. So they spread. So we had to then work on this more. And then the idea of developing the neuronavigator, we start with André, and then Victor came, took over, and then Renan came and took over. And when I see our development, I mean, in terms of methods, I think it's impressive because we had a very crude approach at the beginning. Now we have a neuronavigate system and it's controlled by our robotics. So think about the precision we gain in all these years. But we gain this not without efforts of many people, especially you young guys, students that are there working diligently and putting your time, your efforts to develop that. So now we are even moving forward, right? We want to put the EEG, like Marcio and Victor is doing, to record signals. So I think we are moving forward, and we should not, let's say, give up, because whatever we get in this area, a millimeter of advance, it's important. That's how we are going to understand more. It's not like there will be, in my opinion, a big breakthrough that we will understand everything. No, it'll be like a millimeter by millimeter moving forward situation. So I think you ask a very hard question, but this is my honest answer.
Very nice answer, Professor Buffa. Can I comment on
Sure,
that?
Sure.
So I'm gonna put here in the chat the reference of the publication that I was talking about, the International Federation of Clinical Neurophysiology. I think that can help a lot of people. It summarizes. It's a very good summarize about the use of TMS for diagnosis. And one thing that I'd like to comment is, when we think about diagnosis, we don't think about the other applications of biomarkers, right? Because biomarkers can be used for diagnosis, but also can be used for prognostic values, and also can be used as a surrogate outcome, and also can be used to predict the response to the treatment, right? So I think the main potential of TMS is for these other applications as a biomarker. For instance, in clinical practice, how do we know if the patient are respond well to treatment for pain? We ask questions, we use clinical scales, but we don't know what we are inducing, what type of changes we are inducing in the brain. So a way to do that is to use a periodic pulse and measure GABAergic activity, for instance. And it seems that chronic pain is related to the decrease of inhibition in the brain. So that is not well established yet. But if you get more with the specificity and well established, it has the potential to be used in clinical practice to guide the intervention. Another example, for prognosis in stroke, we use TMS to see if the motor cortical pathway is intact or not. If you do the stimulation, you see the contraction, we know this patient has a better prognosis in comparison with the patient that has not contraction, for instance. And if you see in this publication that I put in that I put there, you will see that TMS is already considered validated for several indications, but most of this indication we don't use in clinical practice. For instance, is validated for Alzheimer's, is validated for multiple sclerosis, but in clinical practice, it's better to ask for MRI. You have a better information of the patient. So that's my main comment. We need to think about biomarkers not only for diagnosis, but also for these other indications of biomarkers.
Thank you, Marcel. Now, Julio, please.
Thank you. My question is related what you were commenting. Now we are starting to have a big gap in technology. For example, in Finland, we're also here now, we have this like robot, we have multilocus, we have DTI, we put everything together, which I think is the way to go. But as a reviewer, many times I receive papers to review where they don't even use neuronavigation. They record the MEPs, and they do some tasks, but they don't use neuronavigation. And I have problems because, I am familiar with all this new technology, all new things, and I think they should be implemented. So the question is for Riston, but what should I do? Reject the paper only because they don't use neuronavigation? I know that it's not accurate because there are a lot of error, right? But what shall we do? And this happens a lot in main groups that they start to work with TMS. They don't have access to all this technology. And I think we are going to have that problem more often because now you're going to be publishing paper where you have everything, but then we are going to have people that they don't use all this like DTI, right? The field is advancing, but we are starting to have this gap in technology. So how to proceed in that case? Thank you.
Well, if you're reviewing a paper, I think that you look at the claim, what are they claiming, and then you look at whether there's evidence. If the evidence can be obtained without neuronavigation, that's fine, and you should accept it if it's an important finding. But there are many things where you need neuronavigation. Then, well, it depends on whether the claim is supported by the data. I think it's simple. It's the same thing with many other things. We had this discussion with EMG. In some cases, you do not need to define it precisely. But in some other cases, you do. It depends really on the question that you're asking.
My comment was along the same direction as Ristos because based on my own history. So when we started, we are at the beginning of the learning curve. So it's kind of natural that you are going to do things that are kind of simple. You don't have all the means, but you should persevere, right? Because otherwise, if you submit the first paper and it's hammered, okay, let's work on the critics and the sendback. And that's how you move, because if you reject the paper and the person also give up, then probably he or she won't receive any funding anymore. And the research is dead at that place. And we don't want that. So we want people to progress, to thrive in a honest way, like Risto was saying. So if the paper is consistent, the claims are correct, the methods they use, even though they are limited, are right, publish it. And let's the community evaluate, right? I won't put any, let's say, obstacle to that. But there is a learning curve. Everybody knows that. It's something different when you have, okay, you send your best student to the best place. He stays there 2 or 3 years and come back. And then he transfers the technology to here. The risk is that the student's so good that he stays there and never come back. And then you are stuck with your situation. So just work hard. That's my point. So any more comments?
Professor Bafa,
Paulo.
até aproveitar.
Marcel, por favor. Sorry.
Eu vou pedir desculpas, mas eu vou ter que sair.
Ok, ok.
Tá, mas foi um enorme prazer participar, viu? Eu agradeço muito a oportunidade.
Tá bom. Obrigado, Marcel. So thank you,
Obrigado.
Marcel, for being with us up to now. He has other issues to deal with, and we are almost at the end. So thank you, Marcel. Nice to have you here also, and I think it worked. Good, good.
Thanks for all the comments. I think I have a comment and a question. Okay. So a long time ago, I followed the work of a researcher called Victor Girsa. I think this. He was using EEG, fMRI to build, I think, a virtual brain in order to simulate how the epileptic seizure propagated around the brain. And his objective was to identify the location of the onset of the epileptic seizure. Okay. What I would like to know is how you see, for example, a approach like applying a TMS and see the EEG produced in tracking this onset of the epileptic seizure, and how this approach can be compared to other ones.
Well, I don't know if you want to comment.
That's what we want to do. I don't have an answer, but we have very good scenarios to test this, because we could. That's, in fact, it was a project that we start to implement during the COVID era. We couldn't have patients, but for this proof of concept, we need, first of all, patients that we have very convergent functional and structural data, that we have electric source imaging, structural damaging in the MRI. We have, so it's all data is convergent to a situation, and that's the cases that usually we do surgery, and we have we call the very good outcome. It's angle 1, patient is seizure free. So that will be a situation that we can have a structural, and we have everything, and then we can use TMS to see all evoked EEG responses during that situation. So when we have a different group that we can see MRI negative patients, so that we don't see most, up to now, most of very successful surgeries are the ones that we see MRI lesions. So when we see like a cortical dysplasia, or a very like a low-grade tumor, or a scar, that's the one that we don't need very sophisticated EEG or depth electrodes approaches, so it's easy. So that will be our control structural group. And then we have the ones that is the more difficult, the most difficult cases in every epilepsy surgery center, is the ones that we don't see any lesion. So that we can use EEG to use as a approach guided to do the TMS. Like we can see the spikes, electric source imaging, functional MRI, and see if we can, in these non-lesional cases, and we see this pattern that will be similar or not. But we don't have the answers. So we can design experiments. We can wait for the ideal patients. We know that patients that are with that very deep structures in the mesial or in very profound structures, they are not good. So I think that Danilo will comment on that some challenges of DMS addressing deep structures. But I think we expect to see data that we can analyze it and use to understand and to add information what we already have. So with the traditional imaging and EEG and other tools.
Okay, I think we had a very good exchange of ideas. I don't know, does anyone know what tricorder is? Tricorder. Do you have any tracker here on the audience? Have you seen Star Trek? Okay, in Star Trek, the physician will come with an equipment and just pass near the person and say, well, you have this and that. So we don't have a tricorder yet, but I'm sure when we have one, it will be magnetic. Because magnetic fields are the only things that can pass through the body and convey information. So take a look at this series, you see the Dr. Spock there, with his ears and so. All right. Okay, so thank you for being here and exchange your ideas. Thank you also for Marcel that are left. And now we have a coffee break and we can continue to exchange ideas in a more informal way, okay? Thank you. Você lembra do tricorder, não?